Welcome to the conference on DNA methods in environmental monitoring and management. My name is Sarah. I will guide you through this conference. And this conference is a collaboration between the Swedish Environmental Protection Agency and the Swedish Agency for Marine and Water Management. We wish you all a very, very welcome. This has been a big interest in this conference, and we have 280 participants, more than 280 participants, and it's a pleasure to be here with you today. I will kickstart this conference, and then we will hear some more from our general directors from the two authorities. You have already started to go in on the method that we will use for contacting you, all you who are listening out there. It's called the Mentimeter, and you go to menti.com, and you will see the Menti number, which you will then check in with. And in there we have some questions for you. So if you haven't been there yet, please start to go in there. The purpose of this conference is to spread knowledge and to meet each other, although during these circumstances we will do that in a different way via this conference. And we will also try to produce some new knowledge in these discussion groups. So we would like you to encourage you to contact each other afterwards. If you need to have the contact information for someone, you can contact the Swedish Environmental Protection Agency, and we will give you the contacts. The technique we are using is that you are watching us via YouTube, and we have our speakers with us in a conference room. And when you are going out in these discussion groups in the afternoon, when we are going to discuss with each other, we are going to do that via Skype. So yesterday you will have gotten an email, and in this email you will have the links for these Skype rooms. As I said before, you go to menti.com, and there you can also afterwards write your questions to the speakers. We will collect the questions, and then we will have a gathering for all the speakers and ask those questions to them. Our main organizer, Gunilla Eidung, Senior Research Officer here at the Swedish Environmental Protection Agency, she will come up and read the questions together with me if we have questions. So, we are checking out the Mentimeter. We had a question to you, and remember during the conference to refresh your site. Every time you have a new question, you need to refresh your site. So you should have all gotten also a link to the website for the conference yesterday. And there you can find the program, you can find links, and you can also, after the conference, find the presentations and notes from the workshop sessions. So let's check out Menti. How are you doing today? We had this morning question about how it looks. We can sort of see that most of you are up and about. We are some of you need a bit more coffee, I see, but most of you also have never been better. So we're happy that you're enjoying this kind of discussions with us and information and communication. Well, we also want to ask you where you're at. And where are you? We know we are in Sweden right now, here in Stockholm, at the Swedish Environmental Protection Agency. But we also would like to see where are you in the different countries. We have not only Nordic countries with us. We know that we also have, for example, Germany watching. So please go into Menti and write down where you're from so we can see. We are going to get a cloud from that. And remember, as I said, to refresh your site so that you get the new question. And we are soon going to speak to Björn Risinger, and Björn is our General Director at the Swedish Environmental Protection Agency. He and Jakob Granit, General Director of the Swedish Agency for Marine and Water Management, will start up this conference and introduce this theme that we are having today. So let's see, do we get any answers on where people are in Mentimeter? I will see if we can get a picture of Mentimeter and see how it looks. If you go in on menti.com and the number to menti.com, you have to sign in with a number, and the number is 1335135. 13 35 13 5. I almost sound like the Eurovision Song Contest. So you will see now the cloud starting to get up. So let's see the cloud in Mentimeter, and I will need to see as well where you all are from. A lot of people are from Sweden, but we also have Switzerland and Germany and China, and it's moving all the time. Norway, Finland, Denmark. Welcome, all of you. Latvia. Nice. Öland, part of Sweden, I think. Very nice. Okay, I think we are going to, while this is working, we will see it, we can share it in the end as well to see how it looks in the end. But we're going to let in our first speaker today, and that is Björn Risinger. A warm welcome to you. You are the General Director, I'm sorry, for the Swedish EPA, the Swedish Environmental Protection Agency. So, a warm welcome to Björn Risinger. We need to have him in the picture, but maybe soon he will get to see us as well. Maybe we can put up there you are. Yep. There is Björn. Welcome, Björn.
Thank you, Sarah, and good morning, everyone. So, welcome to the conference on DNA methods in environmental monitoring and management, arranged by the Swedish EPA and the Swedish Agency for Marine and Water Management. We are really happy that there is such a big interest in the conference, and we appreciate to have participants both from Sweden and from our Nordic neighbors, and many more, as I saw on the Menti screen just recently. The conference is focused on DNA methodology that is used to support environmental management in terrestrial and aquatic environments, with a special focus on environmental DNA to reinforce monitoring of the state of the environment. Personally, I once began in the Nature and Youth Sweden, Fältbiologerna, or in Danish and Norwegian, Natur og Ungdom. Being a nature detective back then was about spending many hours outdoors bird watching, tracking species, or making other kinds of sampling. Those were important and interesting days, but the efficiency and accuracy of the documentation was not always in agreement with the efforts invested. Today, such information could be captured in one single sample. The development in utilizing DNA methods has certainly been a game changer. The advancements are rapid, and we are happy to keep on learning and exploring this area. The core during these 2 days will be research and management in Sweden. We also much appreciate the opportunity to learn about ongoing DNA activities in Norway, Finland, Denmark, and Iceland. We will also learn about ongoing research, management issues, as well as being able to debate on if and how the results are or may be important tools in management. We are all aware of the necessity of environmental management and actions being upheld by best available knowledge. Today, it's becoming more and more important to make it clear that we need knowledge based on scientific facts. Through research, environmental monitoring in collaboration, we create a dynamic and solid knowledge base to be used in our environmental work. Development of new DNA technologies to support monitoring and management is rapid, and DNA is used in more and more subject areas. DNA technology is, for example, used in management of wildlife, both for species identification and counting of individuals. DNA is collected and extracted from droppings, and there are promising results even of DNA extracted from animal tracks. The soil inventory monitoring program uses, for example, environmental DNA from humus samples to monitor the soil fungi species diversity. There are promising indications that environmental DNA will be used in many more ways in environmental monitoring and in management of species and nature. Within the European Union, a joint effort is launched to enhance monitoring of pollinators, where DNA analysis is considered as an important future method. The Swedish EPA is both a funder of applied environmental research and nationally responsible for environmental management and monitoring for policy implementation. And the environmental research grant that the Swedish EPA is managing makes us a unique research funder in Sweden, as this grant is focused on environmental issues in support of environmental policy development towards achieving the Swedish environmental objectives. In 2018, we announced the research call DNA Methods in Environmental Monitoring, and the funded 8 projects will be presented today. That's really nice. We are a curious and interested research funder. We hope that this conference will contribute to increased knowledge about research issues and activities related to environmental DNA, DNA and wildlife, interest-specific variation and management. Moreover, we anticipate that there will be something of interest for the environmental work carried out in all Nordic countries. We also hope that you will get the opportunity to meet people you already know, and that you will be able to make new contacts these days, even though it is a digital conference. And always remember that cooperation and research makes a difference. Thank you, and I wish you 2 fruitful days.
Thank you, Björn. We will also hear from Jakob Granit, the General Director of the Swedish Agency for Marine and Water Management. Jakob, welcome.
Thank you so much, colleagues, and also from my end, welcome to the conference on DNA methods in environmental and wildlife monitoring and management. As you know, we have to do it today in 100% digital form, but I am sure this will be a pleasant surprise for us working in this manner. The conference, as you have heard, is organized jointly by the Swedish Environmental Protection Agency and the Swedish Agency for Marine and Water Management. I'm also very happy that we have colleagues from all over Scandinavia and globally joining this conference with its impressive program. So my name is Jakob Granit, and I'm Director General for the Swedish Agency for Marine and Water Management. And our agency is the responsible central government agency tasked to protect, restore, and ensure sustainable use of freshwater resources and seas, including fisheries management. Monitoring the state of the environment for management actions is critical, but it is, as we all know, difficult, complex, and expensive. However, our management decisions must rely on scientific evidence to the greatest degree. In Sweden, we have over 300,000 kilometers of waterways and about 100,000 lakes. and also almost 48,000 kilometer long coastline and 3 sea areas. And that, of course, is much to monitor in the aquatic environment. So as the responsible agency for aquatic environmental monitoring, this research conference is therefore of utmost importance. We are glad that this conference is arranged in the middle of the joint research program that we announced in 2018, DNA Methods in Environmental Monitoring, supporting both agencies so that we can listen to and take part of early results of the research projects, but also share knowledge in this field in general between ourselves. Returning to the complex task of environmental monitoring, including fish stocks and fisheries, this task is carried out by many agencies in Sweden. And the Swedish Agency for Marine and Water Management is the responsible agency at the national level. But other central government agencies monitor freshwater resources and seas within the respective fields. The country administrative boards direct regional work. Municipalities, companies, and individuals monitor the aquatic environment locally. Higher education institutions and various institutes carry out sampling and analysis and provide research expertise. An important part of our mission is to describe, analyze, and visualize and report on the environmental conditions within our area of responsibility. The information is used when we follow up on how we comply with, among other things, the European Water Framework Directive and the Marine Environment Directive. So from our perspective, we see that eDNA research and development presents innovative tools for studying the genetic diversity of species within and between populations to a degree not accomplished to date. The next major step will then be to choose to find out techniques that can be standardized and include as routine management tools to monitor genetic diversity in relation to major environmental problems, and then of course to guide actions to tackle those problems. There are many promising applications that can be explored moving forward. In 2020, we began a few pilot schemes to monitor genetic diversity in the aquatic environment. The pilots have a limited number of target organisms such as cod, herring, salmon, and eelgrass. And we are very grateful for the work of Gothenburg and Stockholm universities, including the Swedish University of Agriculture and Sciences, who contribute to this work in parallel, in collaboration. We are hopeful when we see projects such as the e-pike that potentially can provide a tool to monitor the situation of the pike stocks that we do not monitor today on a regular scale. As we move forward with the national program to make hydropower sustainable in Sweden, a 20-year-long enterprise, we also see opportunities to use eDNA as a tool to monitor change in connectivity in the waterways that we are addressing. A generic issue to consider is how data is stored and managed in safe ways to promote reuse of the monitoring data for different applications and generations to come. So in conclusion, we are very happy to see so many scientists and practitioners interested in the field of eDNA. That in itself demonstrates the interest to move forward and innovate in the field of environmental monitoring using new technologies. So with those words, I really wish all of us a great 2 days, and thanks for inviting me.
Thank you very much, Jakob. Thank you. Okay, so we have heard our general directors speak, and now we will go to the keynote speaker of the day. We have Professor Torbjörn Ekrem with us. He's at the Norwegian University of Science and Technology. He is professor in biosystematics and a coordinator for the Norwegian Barcode of Life. Welcome, Torbjörn, and over to you.
Thank you very much. And thank you very much to Gunilla and the rest of the committee for inviting me to give this talk. I've been working with barcode reference libraries for quite some time, and it is an exciting time. You might think that we are all sorted out, but as you will see, there are still challenges to come.
Can I just say that you need to hold your microphone? It's swaying against your shirt there.
Oh.
Thank you.
Okay. Sorry about that. All right. So down to business, I guess. DNA barcode, I guess it's a bit
not necessary to explain people here what DNA barcoding is, but nevertheless, it can be really trivial to identify species. Everyone would see that this is a sheep. So DNA barcoding is not really efficient way of looking at the big diversity, the diversity that we all more or less recognize from childhood. But it's extremely useful if you want to look at the true diversity, the diversity that's consisting of fungi, of macroinvertebrates, of insects and so forth, or if there are parts or even traces of that diversity in the environment that you're sampling. And for all of these cases, it will be extremely important to have a good quality reference library to compare your unknown sequences with. If you don't have that, you will not be able to put a name on the sequences that you have retrieved from that environmental sample, for instance. And if you look at the literature and the publications using metabarcoding, eDNA, or DNA barcoding over the years, they are increasing more or less exponentially still, and now amount to thousands and thousands of papers over the last 10 years. So the importance of the reference library is obvious. And it starts off when we generate the reference library by selecting and collecting a specimen. And the big advantage of some of the major reference libraries out there today is that they have the collection data, a photograph of the actual individual, together with the sequence information and all the the trace files and the metadata from the sequences available in one and the same database. As we will see a little bit further down in my talk, this is important if we want to quality control and quality assure the reference library used for identification of species. And the key here is really the specimen, because without the specimen, you won't have any DNA sequence, I guess, but also without keeping the specimen as a voucher for future reference, you also won't have the possibility to go back and look at the identification when the next taxonomic revision occurs, or if you suddenly detect that there is a misidentification or a potential cross-contamination during the laboratory process. So keeping a voucher specimen for future reference is extremely important. Last year, we worked together with the working group on reference libraries in the Cost Action DNA Aquanet. We worked on looking at the gap analysis of aquatic taxa in current barcode reference libraries. And we'll get back to more of those gaps a little bit later in my talk. But I wanted to stress this figure here because we also looked at what's needed for quality assurance and quality control of a reference database. And as you see here, there are basically three different phases in that process. First of all, development and the creation of the reference database, where the quality assurance really is important to consider, such as valid input of the taxonomy, that we follow the formal barcode standards, that we make sure that the voucher is deposited in the correct way, and that we see and make sure that the specimen data is available and retrievable from that database. Then during the lifetime of the database, it needs maintenance. It needs to be updated according to current taxonomy. It needs to be made open and transparent, and it needs to be able to track the different versions of those reference libraries in case a future reference library give a different result of your analysis than the current reference library does. And then, of course, what we will hear a lot about over the next couple of days, how are these reference libraries utilized? And the quality assurance there is, of course, important for the utilization part of reference libraries as well. For instance, that we can tag the type material in there. We can flag if there are discordances and make sure that the different parts of the libraries that are used are tagged with digital object identifiers, DOIs. So right now I would like to talk a little bit about more quality assurance and then get back to more the quality control part later in this presentation. CBOL, some years ago, the consortium of Barcode of Life, that is, as part of the barcode community, set up a number of requirements that should be met in order to get a formal barcode status of the sequences deposited in the repository. There were something like a unique voucher identifier so that you can actually retrieve the voucher afterwards. It should be an identification to species level or a provisional label for unpublished species. A country code was needed, an accepted barcode region in the submission, which means that they need to be from one of the accepted genes, depending on which organism that you are looking at. And at least 75% continuous high quality base calls for that particular reference barcode. For CO1, that's roughly 500 base pairs. And it must be associated with trace files in the repository. So this was generated in the time where there was only Sanger sequencing and no high throughput sequencing technology available. So, naturally, these requirements are kind of mirroring that. And then the primer sequences are needed to be published along with the barcode itself. So these are all good, but you see that there are quite a bit of focus on the molecular part of these barcodes, and not so much maybe on the voucher part or the specimen part as requirements. But there were some recommendations in addition to those requirements. For instance, you should provide a latitude and longitude, a name of the identifier, name of the collector, and a date of the collection. And those are, of course, also important parts to be able to associate the identification with the current taxonomy. And maybe even the date of the identification should be an important place in here, or you think where it actually is stored. So we think that there were, these requirements that are not really, and recommendations are not really sufficient to optimally assure the quality of a barcode reference library. So as part of the DNA Aquanet cost action, several of us in Working Group 1 have been collaborating on writing up a standard paper on metadata and practical guidelines for specimen and DNA curation when you build a barcode reference library of life. Because we see that not all barcode, what I call reference libraries, are actually meeting the standards that should be required. For instance, what about the storage of voucher specimens? They should be placed in a publicly accessible collection. How has DNA been harvested? Can that influence the downstream analysis? Or how has the DNA been stored? Is the DNA stored along with the voucher specimens? Can it be used for future DNA analysis of additional markers, maybe to test taxonomic hypotheses? And the metadata, both belonging to the specimen itself, its taxonomic information, the identification made by researchers and specialists, and the vouchers, and also metadata belonging to the DNA markers. What kind of instrumentation was used to generate the DNA marker? What kind of primers were used? What kind of quality scores can be associated with the DNA marker and DNA sequence that is present? So to dig down a little bit in the voucher issue, which is my heart close because I work in the museum maybe, and I've always been looking at vouchers and keeping scientific collections for my research. Here we can look at both the storage of vouchers and this beautiful beetle here, for instance, that was just leg picked for DNA barcoding at our museum. It's still stored there in the museum with an identification number unique to our collections, and where it is also available for examination and loan by fellow colleagues. maybe someone doubt the identification that was made and would like to take a look at it, or include the specimen in a future taxonomic revision. The storage condition of which how we store this particular specimen, you can see already that it's glued onto a cardboard and that is pinned. And this is how we store a lot of our insect collection. But for other organism groups, for instance, diatoms or vascular plants or fungi, they are stored in completely different matter. So in our paper here, we are also looking and proposing some optimal storing conditions for specimens, both so that they are kept well for future reference of morphological identifications, but also so that they are kept in a way that will not prevent at least future DNA analysis. And this voucher can be a digital voucher also, if it's impossible or difficult to preserve the actual specimen. We have issues like that with, for instance, rotifers or tardigrades, really microscopic organisms that when you sequence or extract DNA from them, there's not much left to take care of. But then if we are careful and make a proper high-quality digital image of the specimen before DNA extraction, we can keep that as a proxy of the voucher. In terms of the metadata, we are of course interested in locality information, both coordinates, country, and the name of the locality. We would like to keep the date in a format that's recognizable by all computer reading softwares. And also the collector, a photo of the specimen, and the preservation status of that specimen, so that we know if it's, if it's part of the specimen that's available or if it's the full specimen itself. And it's also important to know how you can access that specimen. Is it out for loan? Do you have to go there to visit it and to examine it? And especially during these times with very restricted travel, it's not that easy to access in many cases. So important part, as I see it, in the storage of voucher and voucher metadata. And in our paper, we go through many different aspects of keeping vouchers and the metadata standards. In terms of gaps in the reference library, this was a really nice activity that we did during this DNA Aquanet cost action. Here we looked at which species are used for biomonitoring of aquatic habitats in Europe. So we looked at both marine environment and we looked at the freshwater environments, and of course also brackish water. And you should think that many of the European countries look at approximately the same species in each of these groups, but they don't. So our first exercise during this work was to actually find out which species are monitored in which countries. But when we had that information, it was really interesting to use that and to compare with the coverage of reference libraries at the same time. So if you look at the AMBI or the AMBI marine invertebrates, those are marine invertebrates that are used to generate the AMBI index, and that's frequently used in marine biomonitoring. And this graph here on the left has two panels on the left and on the right side. On the left side, we see that the barcode coverage of different organism groups that are represented by at least one barcode per specimen or more. And in terms of some species groups here, like the decapods, for instance, they have a very high coverage, more than 80%, but there are only 25 species actually being monitored all over Europe of these decapods. While for the mollusks, for instance, we only have about 50% coverage in the barcode library of those 625 species that are covered. And then you see different colors here on the bars. So the dark blue bars, they represent the public data available in bold, while the light blue bars add the private data available in bold. And we have added that because it can be used for identification when using the bold identification engine. And then the gray data also includes data, or the gray part of the bar also includes data from GenBank. So you can see there that there is a considerable lower amount of the barcodes available if you go for the high quality public and open reference library in bold. In the right panel then, you see the barcode coverage if there are at least 5 barcode references per species. And we use this as a kind of a quality measure, considering that more barcodes per species would be a more secure, more higher quality reference database than only 1 barcode. And here you see how the coverage falls drastically in most groups, and the mollusks now are down to about 30%, a little bit less than 30% in total. And if you look at the bold public data, you're down to almost a little bit more than 10%. So a big difference there. When we know the coverage of barcode in barcode reference library, this is important because, you know, if it's not in the reference library, you will not get a hit on your unidentified sequence. If we focus in here a little bit and look at the freshwater mollusk, just for a comparison, we see now that we can use the country boundaries to see the representation of each of these species in the barcode reference library. So the first panel up here to the left shows how many species are monitored in the different countries. And the darker blue colors represent more, indicate more species. So the Czech Republic here has a lot of species that they are monitoring for their freshwater biomonitoring programs, while countries like Finland and Italy has a much lower number of species available, at least in the lists that we got. The first panel to the right here, panel B, shows the barcode coverage as the public data in bold with at least 1 barcode reference. And then you see, as we have the total, including the private data in panel C, and the total data in panel D for GenBank, the coverage increases quite drastically. In panel E and F, we see how the barcode coverage with a total limit of 5 barcode references per species is in total, and then including GenBank in F. So there you see the coverage goes down a little bit, and this is what expected. Another interesting thing that we looked at this in this study also was which countries and how many countries have monitor each of these species. So here you see that the number of countries on the lower part of the panel, and then you see the number of barcodes for each of those species. Each column is a separate species. So it's kind of, I guess, comforting to see that at least for the species that are monitored by many countries in this part of the graph, there are less gaps in the barcode library than for the species only monitored by 1 country. So over here in the right part of the graph. But still, there are quite a few gaps in between here that can be covered. And we see that there are quite a few of these species where that are only represented in the private database or in GenBank, the gray bars. So what happens then if we don't have a well-represented library? Well, obviously, if you don't have the species in there, you will not get a species identification on your sequence. But can you get the genus identification or a family identification of that unknown? So can you at least get a little bit along the way? We tested this on chironomids, now it's 13 years ago already, with a rather small data set, and found that we needed only, just by looking at our own small data set, we could say that we need a comprehensive DNA sequence library if we want to identify species by DNA barcoding. And the bottom graph here shows that we had quite a nice representation in terms of interspecific versus interspecific divergence. We would like to see that there is no overlap here, and there actually was no overlap, although the outliers were fairly close, as you can see. But if you look at our neighbor joining tree, we had some interesting results. First and foremost, we had several perfect matches, even though they were specimens from very different areas of the world. So for Paratonytarsus grimi, the perfect match down here, the sequences were identical. That was nice. We also had some instances that indicated a cryptic species. So morphospecies that obviously were genetically, where populations were genetically very, very divergent. But then we also have some indications of unknown. So single branches in this tree where you cannot really say which genus does this belong to. And we cannot say that because the genera in this tree are not monophyletic. You see Micropsectra, this is what's large clade on top here. It's not monophyletic. We have Micropsectra here. We have one Micropsectra here. We also have Micropsectra down here. And now we know also that Parapsectra actually is part of Micropsectra. So these clades say that, okay, if we get a branch of an unknown sequence that ends up at this particular position in the tree, we cannot really say if it belongs to Micropsectra or any of the other genera represented in this particular data set. So that was one of the outcomes of this paper, and I just wanted to see, well, what now? 13 years after, we obviously have a lot more sequences in the database, and you should think that when you start populating the database and it gets very close to completion, that many of these gaps will be filled, and that the genera will come out as monophyletic groups, and that a proxy for identification to genus level, for instance, might be possible for these chironomids. So I did a small test here the other day, looking at almost 20,000 records of chironomids of the subfamily Chironomini. But when I chose only those with more than 600 base pair CO1 sequences, I ended up with about half of that. So about 10,000 sequences, 687 species, and almost 1,200 bins. And I'll come back to why there is the discrepancy there. Just a simple neighbor joining tree, but it ended up with 100 PDF pages of trees, so we won't go through all of those. But looking at one page or part of a page that very nicely illustrates that genera are not monophyletic, even if we add a lot of data to the Chironomid barcode library. You see here all the Tanitarsus groups have different relationships all over this tree. The green letters here represent the tribe Tanitarsini, while the purple letters represent the tribe Chironomini. And you see that not even the tribe level there is any monophily to be suggested in this figure. And these unknowns then, here are two that has only been identified to subfamily level. Which genus do they really belong to? Well, it's impossible to say based on this tree. And the reason here is that there are so long branches ending up into species, while the branches grouping species together down here are so short with very little support. The marker is just too variable to say something about the high level of relationships in chironomids, and therefore it's difficult to assign genus level identifications if the species is not already present in the barcode library. Okay, another thing that has been debated quite a bit both in the literature and at conferences is what happened if you lack regional representation in your database. Let's say some of these species have fairly wide distributions, and you could have a nice distribution of barcodes from, let's say, North America even, and that same species might be present in Scandinavia. Is it sufficient to have that barcode reference represented from, or that species represented from North America in the reference library? In some cases, it can be quite sufficient, but in other cases, like they seen for these moths on oak trees, there is quite a big difference in genetic distance depending on where you have collected the actual specimen. So what Gaitan et al. Looked at was these various moth species. The green one looked at here is a Teutrix moth, and just as a representation. But what they saw here is that if they looked at the pairwise distances between Central European and Iberian populations, this is a much higher general average level of genetic divergence than if you look at Central European versus Italian, or even Central European versus Central European. So the genetic or the geographical distance certainly has an influence both on the actual genetic distance between specimens of the same species, but also on the uncertainty, the variability, the standard deviation here. As you increase the geographic distance, you also increase the divergence and the standard deviation in those pairwise comparisons. The graph to the right here shows a slightly different story, because here we see that Iberian populations of the same species also show a higher interspecific genetic divergence than European populations of the same species has. It all still increases by geographical distance, but you can see it's considerably higher than what you see in European population or Central European populations. And this is explained or can be explained by hotspot habitats for these particular groups. We've had oak trees and many different oak trees over a long period of time, and has not been really interrupted by Pleistocene glaciations and so forth down in the Iberian Peninsula, while in Central Europe looks very different today from what it was 10,000 or 15,000 years ago. So that's one explanation. But it says something about the need for regional representation in the barcode reference library, because if you don't have that, you can risk getting a poor identification, or maybe no identification at all of your species. And this is also part of the argument why we in NORBOL would like to have sequences from all Norwegian populations of the species that we have in Norway, although the species might have been sequenced from Germany, for instance. So now a bit over to the quality control part of reference library. The first graph here that you saw a bit earlier also, you see that there are some quality control points down in the line here. For instance, you would like to evaluate additional taxonomic information if you find things that are discordant in your reference library. And you would like to curate library entries, or you need to curate library entries during the lifetime of that reference library, because there could be taxonomic revisions, there could be new data coming in that convince you that the identification originally placed in the reference library was perhaps not the best one. Maybe there are synonymies coming in, maybe there are classifications that have been changed, and so forth. And also, as a quality control point for the use of barcode reference library, we need to consider the metadata output so that it can be included. And maybe for regional representation, we would like to define some custom reference libraries that are of exceptional quality for that region compared to on a global scale. One way to control the quality of reference library is, of course, to do proper taxonomic work and testing species hypotheses and phylogenies. So using a multi-marker approach, you can look to see if the DNA barcodes delimit species the same way as a more extensive and better argued data set does. In this paper here, again on midges, you see that by using multiple markers, we could confirm that the barcode data for this group of species represent the morphological species quite well. There was only one instance, and that's Tanitarsis brundini that you see in the olive green one here, where you have one population in Norway that is quite different from other populations in Norway. But morphologically, no difference at all. And for nuclear markers, there were no differences either. So in this case, these two are seen as very divergent, genetically divergent populations of the same species. But this is a very tedious way to go about if you want to quality control your reference library. Can you imagine doing this for all 10 million species on Earth? That will be quite a task, and we will never reach that goal, I guess. But luckily there are other ways where you can do some very, well, you can call it quick and dirty, but it's quick but not necessarily so dirty, I guess. For instance, the UNITE database for fungi have a fungal species hypothesis incorporated as part of the database. So in that database, you can go down here and you can test the various thresholds of intraspecific genetic divergence. And you can look at your particular group of interest with a particular threshold. And you could see there if your unknowns or your species database, how that relates to morphospecies and the identifications that are available in the database at different genetic thresholds. And that is a very useful proxy or comparison to do, because if you have some genetic clusters as species hypothesis, then okay, you can go in and see where there is discordance and investigate that further. In BOLD, you have something similar called the barcode index numbering system, BINS. And these are also essentially species hypotheses that can be used to see if the taxonomic identification by various
Torbjörn,
authors
we have
in
contact.
the database
Can we just
belong to the same genetic cluster.
Torbjörn?
So here's another chironomid. Beautiful, isn't it? We have about 118 public records for this one, and you can see that there's a maximum distance of 3.7%.
Torbjörn, can I ask you? Can you just hold your microphone again, so
Sure.
that it will not swing on your shirt? Thank you.
Okay, thank you. Well, in talking about chironomids, you get so eager, so that's why I forget. So back to this. Could these are the bins then good proxies for species in these non-biting midges? Well, here you see an indication that it might not be. The blue bars here indicate within bin genetic divergence, while the red bars indicate the distance to the nearest bins. And you see there is a quite large overlap between species here. Well, not between species, between the bins. And this is very indicative of what we've found in all our studies on chironomids where we investigated this. We have to expect about 4 to 5% divergence within species for chironomids, and that's much higher than we see in many other insect groups. So bins do not correspond very well with morphospecies. We have many more bins than we have morphospecies in BOLD, and whenever we investigate that, we see that it might be that definitely is a bit overrepresented compared to the actual morphospecies. And one explanation for this could be that we have had large population sizes over very long time periods, and this is certainly the case for many chironomid species. So it's of course quite tedious if you want to go through every single bin and look for any discrepancies, and there are tools both in BOLD and other places that can help you along. One that was recently developed by some Portuguese colleagues called BAGS is an automated barcode audit and grade system for DNA barcode reference libraries. So these are focusing on animals where we have bins, and by using a library selection or a list of species, you can filter that towards marine taxa if you'd like, but you can also mine directly in bold and get out all the bins and records of these species for auditing and annotation, and to look at bin versus species name congruence. When you have done that, you can then put different grades on these bins to see, which one of these are in 100% correspondence with species and which are probably discordant and needs a closer examination. So the thinking of the whole grading system goes like this. If you have a species that share a bin with other species, then that would be grade A, the discordant species assignment. If not, the next question is, does the species have less than 3 species in the library? Yes, okay, that is considered insufficient data, because you would need to have more than 3 records to say anything useful. So that's grade D. For no, the next question will be, okay, is the species assigned to more than 1 bin? Yes, that's grade C. You have multiple bins with the same species name. And this is something that we see very often in chironomids. No, the next question will be, well, does the species have more than 10 specimens in the reference library? No, it doesn't. So there's a basal concordance there, but it's to have more than 10 records. Yes, this will be what we call a consolidated concordance, a grade A. And as an example, I like to look at Chironomids. This is then the genus Tonitarsis that we've been talking about earlier today as well. Running that through BOLD and the list of species for all BOLD records, we see that in the graph plot and the plot here, that the number of specimens per grade is quite high for grade C. So we have many specimens that of the same species that belong to multiple clades. On the number of species per grade, we see that there is a quite different distribution, and a bit alarmingly maybe that we have quite a few bins that have multiple species names. And the reason for this can be many, it could be the difference of opinion of the specialists that have been identifying it. It could also be that 1 or 2 records in that bin has an interim name, while the rest has the true name, and so forth. But we see also that there is a large group D and a quite large group C. So there's still quite some work to do in curating the barcode reference library for chironomids. And the good thing with BAGS is that you can then download a list of all of these categories, and you can use that to go directly into the bins and edit and refine the taxonomic information that's available in the database.
Okay, you have now about 5 minutes left if you want to use them.
That's perfect. So towards the end now, I would like to talk a little bit about FAIR principles, because if a library is not available, it's not findable, it's not transferable, it's not interoperable or repeatable, it's very difficult to repeat the study that has been done. And that could be a research study, but it could also be a monitoring study. If your reference library is not available, it's difficult to repeat it. So I encourage people to make the reference library machine readable and to make it available and accessible through public repositories and the use of DOIs. So what about the future then? What will the reference library, what are our challenges, and what can we improve with our reference library in addition to what I've been talking about already? Well, the first thing I would like to see is that for some organism groups, we've tried to increase the number of markers that are available in the barcode reference library. And we've seen that this is very useful for vascular plants, but also for marine invertebrates that seem to be notoriously difficult to sequence for CO1. We could use genome skimming to assemble the full mitochondrial genome and extract the barcode markers out of that. And then they will be better to complement the reference library. And people looking at the monitoring studies or community studies in the marine habitats can also use other markers than CO1 in their study. This has been done for plants already, and the group by Inger Alsos in Tromsø had a very good and high success on using herbarium specimens and genome skimming to provide barcode references for the Norwegian vascular plants. The other thing that I would like to see in the future of reference libraries is symbiome information. You know, no species is an island, and there are and all of these specimens interact with other species in an environment, either as food sources or as parasites, or maybe just visiting flowers, having fungi, hyperparasitism, microbiomes, and all of these species interactions are extremely valuable information if you want to monitor and manage nature effectively. What happens if one species go away from your network? Will it affect the other species in that network? In order to answer the questions like that, we need to have more information of those networks out in the various ecosystems. And by including those links proven by molecular identification in a commonly available database, you can actually see in a much larger extent how different organisms depend on each other. So that will be an interesting place to go, and this is also what the BioScan project by the International Barcode of Life has included as one of the main targets. So to finish up with this beautiful image from the Trinity College in Dublin, species identification using molecular tools are only as good as the reference library. So if we have a poor quality reference library, we will get a poor quality identification. And therefore, the quality assurance and quality control is really crucial. Know what you have, and the quality of the content is extremely important. If you're looking for a word in this library at the Trinity College and you don't have a good system of finding it, it will just be lost in there. So we need a good system, and we need good descriptors, the metadata. Libraries also need to be repeatable, or studies need to be repeatable, and libraries must be accessible in order for that to happen. Okay, thanks everyone. Just in the end, I would like to thank all colleagues in the NORBL network and the Cost Action and DNA Aquanet for very fruitful discussions and very good collaboration when it comes to developing standards and quality assurance and doing gap analysis of barcode reference libraries. And also the Norwegian Environment Agency, Forskningsrådet, and the Norwegian Biodiversity Information Center for supporting the NORBL work. Thank you.
Thank you, Torbjörn. You have given us a good overview for the coming days, and a good list of wishes for the future as well. Thank you. Before we will go in and hear about these 8 projects, the research projects that Björn Risinger told us about, we are going to take 5 minutes break. So I think some of you want to collect some coffee, and we will be back in 5 minutes. You will see a clock on the channel, and in 5 minutes we will be back. So about 11:07 or something like that. Thank you. 5 minutes.
pilot venture in this painstaking challenge to create
Har jag min
these
mikrofon på?
big reference libraries. And I
Vi har ljud ut. Ja, men då så. Jag tror vi funderade lite grann på tekniken här och vi ska bara testa lite så att vi...
Yes.
Is everything works now? Okay, now everything works. Perfect. Then you can continue.
Do you want me to start from the beginning?
Yes, please do that.
Yes. Thank you, Sarah. Sarah had problems pronouncing clitellates, and clitellata is the proper scientific name for this group. Good morning. I'm Christer Erseus, and I'm happy to be the first speaker in this session on these presentations of the projects of the DNA call. And it refers back to what Torbjörn Ekrem just has been talking about in his keynote talk. You can say that our project is a kind of pilot venture in the painstaking challenge to create barcode reference libraries. And so it's a kind of case study, you could say, in this big challenge. And I will start just by repeating things that we essentially heard already, but it's very important to have a valid taxonomy. I hope that I can provide some of that. I've been working on this clitellate group for 48 years now. And for me, it's very important to have these well-supported species hypotheses. I regard species as hypotheses, and we continuously try to test them in different ways. And the more support we have for them, the better. And also, as Torbjörn said, we need a reference library that is as complete as possible. And it has to be established and managed with good quality control. The problems that I see with the group we are working with is that there is a lot of genetic variation within species, and that this is also something that is becoming more and more evident, as Torbjörn pointed out. One problem is cryptic species, which creates a nomenclatural problem. You have one name, one Linnaean binomial name, but it covers a whole complex of species. So what is the species out of these that should keep that name? So that's one big challenge. I also see species delimitation as a multi-locus, multi-marker approach. It's very important that you use full genetic information to delimit the species. And here, the mitochondrial barcodes are not enough, in my opinion. You need to use also nuclear markers to do this. We also have the problem that when a particular barcode does not work for all the species of a group or for several groups, this could basically be a primer problem, but it is a problem, which opens for the alternative of using other markers as a supplement. So clitellata, 1/3 of the annelid phylum. There are thousands of species known, and maybe 10 times as many in reality. These are the segmented worms that have a girdle, this clitellum, which you can see even in the microscopical form here. This worm is only about 2 millimeters long. The iconic groups are the earthworms. There are many earthworms in the tropical areas, but in Scandinavia, in Northern Europe, we only have about 40 or 50 species. And leeches is another group, but in our fauna, only about 5% of all the about half as many as the earthworms. That is about 5% of all clitellates. But clitellates occur everywhere. They are in the sea, they are in fresh waters, they are in the groundwater, they are in soils of different kinds. So they are very, very common. We know in our project we have collected 550 species or a little more now from Scandinavia. We know that there are many cryptic species, and we have already created species hypotheses which are based on multi-locus information. And here, for instance, is a list of the 18 species of one genus, a genus of small freshwater worms. Basically, they also occur in wet soils. Chernosvitoviella, 18 species in Scandinavia, but we only have 8 names that we can apply on these. And when you look at the list here, even some names are repeated because we know that there are more than one species within a complex. But the DNA barcodes are very efficient to separate all these different species. But at the other end of the spectrum, here is an example of a species, much bigger species. They among the biggest of its family, the Enchytraeidae. They live in soil, and it's called Fredericia magna because it's so big. Here we have very high interspecific variation, and these are only a sample of barcodes that we have from Sweden and Norway. And you can see immediately that there are clusters that are very suggestive of different species, but they are not. They are just one species. Our project, which now has funding from the Swedish EPA, has the aim to establish the libraries of Scandinavian clitellates. And we want to be as complete as we can when it comes to coverage of species and the diversity within species. But we also have the ambition to make libraries actually as for 4 different markers, 2 mitochondrial markers, CO1 and 16S, and 2 nuclear markers, ITS-H3, which is histone 3. The latter can actually be used as barcodes, but in some cases they have problems distinguishing sister species. The base of, or the basis of this big work that we are doing is something that we have accumulated over more than 20 years. We have barcoded about 20,000 specimens of this group from all of Scandinavia, mostly Sweden and Norway, but also from Denmark. And we have well over 550 species, and we are finding a few new ones every now and then still. But we think we are nearly complete as in terms of species. And as I said, we have to also continue to work on testing the different hypotheses we have. This is also in parallel a part of the project. We are just in the beginning of building a database, and we will try to use the Pluto-F biodiversity platform, which has some connections also with Unite that we have heard about in Torbjörn's talk. And we are also very specimen focused. Our 20,000 specimens are numbered. We have sequences from all of them. So we have one specimen with maybe 4 markers and another one with maybe 4, but at least a CO1 sequence, or a couple of the others. And together, these specimens and the research on species delimitation will give us species hypotheses up here. And we also have the metadata that Torbjörn was mentioning. We always have a collection information about where with the latitude and longitude and etc. But we also can use some of this for the other specifics of each species, where we can say something about distribution and ecology. And then we will also include the taxonomy, the classification, of course, but also the descriptions in the literature with the literature references. And we hope to have some pictures to present. And we also want to do some sort of intuitive, at least, quality control about how these species can be used in environmental work, such as if they can be indicators of something. And we know that some species are really indicative of certain habitats and also pollution status. Technically, all of the sequences that we validate as part of these hypotheses will be published publicly in those databases that are there. We don't want to do this separately, but of course they will be linked to with our database. So we also hope to be able to validate other sequences belonging to that we can match with our sequences, so that we can tell which other records in these public databases are trustable.
Christer, you have some more minutes left.
Yes, and this is my final slide. And I want to acknowledge that I'm not alone. I have two excellent co-workers, Morten Klint and Svante Martinsson. I'm also very happy to announce that we've been well supported over the years by both the Swedish and the Norwegian taxonomy initiatives. And I also want to tell you, if you know the Swedish language, which is not that difficult, you can find our YouTube channel using this maskar på jobbet. That means worms at work. And it has a double meaning in Swedish, but that's hard to explain. So please check it out. This is meant to be more like a popular science channel, where we try to make worms popular. And Sara, I know that you love worms. Thank you.
Yes, they are not my favorite animal. That is true. Thank you very much, Christer. We are going to talk to our second speaker, and we welcome Maria Kahlert. Maria is Associate Professor in Biology. She is situated in the Department of Aquatic Sciences and Assessment at the Swedish University of Agricultural Science. Maria will speak to us about barcoding of freshwater taxa for improved assessment of biodiversity, and FreshBAR. We will also remind you that use menti.com for questions to these speakers. And when you have a question, please write to whom, the name to the speaker, so that we know later on what question we should ask what person. Maria, thank you. Warm welcome to you.
Yes, thank you for the introduction. We jump over to barcoding of freshwater taxa for improved assessment of biodiversity, the project called FreshBAR, where we also have some worms and we have diatoms. And I'm the project leader together with, and two colleagues of my department are included, Willem Goedkoop and Richard Johnson. Then the Swedish Museum of Natural History is included with Thomas Lyrholm and Yngve Brodin, and Jonas Zimmermann from the Botanic Garden and Botanical Museum in Berlin and Freie Universität Berlin. Yes, and our aims are, as probably all our aims are, to contribute to the further development of libraries of key freshwater organism groups. And we focus on diatoms, as I said, on chironomids and oligochaetes, and we also check we check those in understudied freshwater habitats. And the aim is to establish and integrate DNA meta-barcoding techniques for these organisms with national monitoring in Sweden. We want to use meta-barcoding in monitoring for real, as our department is already doing with morphological identified organisms. We think that meta-barcoding has the potential to improve monitoring via an improved taxa identification. There are several things that I would like to point out. The first that I'm very much aware of working with diatoms is that with DNA barcoding, we get an automated identification, which hopefully gives us a better harmonization of species identification, which can be a problem in this group, and a better reproducibility. For all groups, I guess, hopefully it's true that we will reduce time for identification and reduce costs in that way, so we can do more. And for other groups, you also get a better estimate of biodiversity, hopefully. For example, chironomid larvae can't be identified to species in many cases, or oligochaetes can't. So hopefully with DNA barcoding, we get better diversity estimates, and then we can develop better bioindicators and new tools. The team, we have two work packages, one for the diatoms, one for the inverts, and in the diatom team also Demetrio Mora and from Berlin and Bonnie Bailey, my PhD students, are involved. And we check diatoms especially from acidic and oligotrophic habitats and alpine regions, because there are not so many in the existing DNA databases. And the invertebrates, those 4 people that I just presented, are making this working package, and they focus on alpine regions and the profundal of lakes, because here you get a lot of red worms, which are difficult to identify. And for the diatom work package, our project plan is that we generate barcodes, 2 different ones, 18S V4 region and the RBCL. Of freshwater only, benthic diatom taxa. It's enough because we have already 1,500 species, at least in Sweden, only in Sweden, so in freshwater. And a lot of them are missing in DNA barcoding reference database. I heard Torbjörn say at least you need to have at least 5 specimens of every species. I will be happy if I have 1 of each one day. We try to use, or we plan to use, 4 different methods. The first is quite traditional, culturing and sequencing clones isolated from a single cell with the micropipette. We also tried agar plates, but the micropipette works better. Demetrio is really good at that. And then do Sanger sequencing. One fancy technique that Jonas is using in Berlin is a direct single cell sequencing of large cells that you can't culture. So you just take the DNA out from one cell. I've never done this, and I wanted to go to Berlin to do it, but hopefully I can in the future. Then we also wanted to do meta-barcoding. We have started coupled to morphological analysis if you have low diversity samples. So you can say, okay, this sequence probably belongs to this specimen. They want to try both with short sequences with established methods, and then you can just get a part of the Sanger sequence barcode, and that's not enough, at least not enough for diatoms to separate species. So we wanted to try long sequence meta barcoding with PacBio to get those sequences. And I'm going to talk about the first one because that's what we have done so far, and where some results can be presented. And yes, we also, of course, want to implement the DNA barcodes and the related metadata into a reference database and establish one. And in our case, it's Diet Barcode, which is an open access and curated barcode library for diatoms. So you can find more about it here. And of course, we also want to meet all the criteria that Torbjörn was just presenting for quality control to be able to link back to the formal taxonomic literature. So we want to also implement vouchers and the metadata to Diet Barcode, and the vouchers to museums, and the physical vouchers should be stored at BGBM, like permanent slice or ACM stubs, DNA extracts. Then I had the plan to send the living cultures, which usually are destroyed because they are not so easy to get in long-term storage, to send to the Diatom collection in Belgium, because they are really experts on this. And the environmental subsamples will be stored at SLU. Yeah, and so here you see what we have in the Diatom database, a lot of references you can think, but for example, here, this is the genus Oinosia. Only 6 species of this genus are in the reference library so far, but we have at least 80 species in Sweden, and they are often dominating, for example, in acidic habitats. So we really have the reference library is covering a lot of Central Europe diatoms, but not really our outpost here. So for example, Oinosia incisa is missing, Oinosia exigua are missing, and they are very dominant sometimes. So some results. Yeah, I was impressed by the speed that some people can work. Thank you, Dimitrio and Bonnie, for this. I contributed a bit also, and we got 200 clones from isolated single cells. And that was the double of what we were planning. The DNA is extracted already. All clones are sequenced for both barcodes already, and all sequences are edited for both markers also. And that's thanks to Berlin, who did all this work, and Jonas. So I can show you some results. It's just very, very fresh. That's how it looks like. So this is we found several new sequences. We managed to get some Oinosia at least, so we can complete a bit. So here you see all the Oinosia, which are nicely aligned. And you can see the first 2 is probably Oinosia incisa, which is missing. The second 2 is hopefully exigua. And then I have some better pictures of the clones of probably Mucophila. And this one, I certainly have to go deeper into identification. I have no idea what it is, other than it's Oinosia. And some maybe Mucophila, but probably something different, and very nice Flexuosa, which probably are not Flexuosa, but something else. We need to go to electronic microscopic identification here. Yeah. And some results of the inwards. The plan is also here to analyze DNA beta barcodes with focus on chironomids and oligochaeta, and to gain better insight into the biodiversity of these ecologically important but taxonomically complex groups, supplement existing libraries with new barcodes by collecting adult midge samples of yet unknown species, and also to couple to see if we can find more species that way. And then also compare the performance of different barcodes, 3 different ones, if you can get a better separation of chironomid species. And also here, the future use of the results will be that we want to make new bioindicators. We want to calibrate the ecological status in a better way if we have a better idea on the biodiversity and the ecology of these species. Yeah, to get better in assessing ecological status. And I'm going to present a bit of these 2 first plan topics. And we have the first results done by the National Museum in Stockholm, and from 12 lakes and the Profondal, so the bottom of these lakes. They were humid, clear, oligotrophic, and eutrophic in 2 different years. Analyzed after bulk extraction with metabarcoding on 1 primer first, and it was a successful sequencing. And most sequences, as expected, could be matched to chironomids and oligosheets, and 57% were chironomids, which I will present a bit more here. We found 78 species with metabarcoding using the BOLT system, and only 17 were found before in the same lakes with morphological traditional methods. So you see already here that metabarcoding really can greatly increase the amount of possible bioindicators. We need, of course, now to check which ecology they have, but it's a high potential for improved indicators for the profundal of lakes. We also found 7 species probably new to Sweden, and 20 species. Back 1 slide, 2 slides.
Yeah. Next steps. There? Next steps.
Okay.
There, you know. Okay. So we'll just have to wait to see if we can go online again. Are we online soon? Can we go? Okay, we lost connection and we try again. Maria Collert has just gone to the next steps. So Maria, please continue.
So next steps. I just would like to say that for the diatoms, I'm going, at least the plan is I will identify by microscopy all these clones at my department. But for the electronical identification and all the washing and also hopefully for the direct single cell sequencing, I need to go to Berlin, and I'm really looking forward to it, and I hope it will be possible soon. And we also will do hopefully be able to continue the metabarcoding with the long sequences, especially at SLU, which also got stopped. And then for the work package 2, we will try 2 more barcodes to see if we can separate species in a better way. We will also analyze more lakes, profile of lakes. The SLU has an archive there, and the plan is to do up to 100. And we will also share the oligosheet sequence that we get with Krista Ezeus, which just presented, to hopefully get more names. And we will do more sampling, which was not possible this year with understudied ecology in alpine regions. And yes, so that's it. Thank you. And there's a lot of people and also institutions to thank to.
Thank you, Maria. We had some connection issues there, but we finally came into goal. So thank you very much. Our third speaker today, before we go to lunch, we will have some questions, summarized questions to all of the speakers before lunch. So our last speaker will be Agneta Andersson. She's professor in pelagic ecology at the Department of Ecology and Environmental Sciences at Umeå University. As I understand, she's also the scientific coordinator for the Swedish Marine Strategic Research Program, EcoChange. And Agneta, you will talk to us about DNA metabarcoding of marine phytoplankton. Welcome, Agneta.
Thank you very much for that.
Exactly. You just have to change. Exactly. Perfect.
Is that okay?
That's perfect.
So thank you very much for the introduction. So, and I will share this presentation together with Meike Latz and Bengt Karlsson. So we are working with a very beautiful and diverse group of organisms. The phytoplankton, which you can see here on some example. And they contribute with, they're very important because they contribute with 50% of the primary production on Earth, and they're very important in the sea. And so therefore they're used in monitoring programs to classify the ecological status of waters. So the project is shared between 3 different organizations. Umeå University, 2 persons, and the SMHI in Gothenburg, 3 persons. So in monitoring, we take sample using a plastic hose, as you can see, when we are on the ship, on board ship. We take samples with plastic hose, put the water in a bucket and preserve the sample with Lugol solution to fix the sample. Then we sediment the sample overnight in sedimentation chambers, and we analyze the sample in microscope. So we do species identification, we calculate abundance, biovolume, and carbon biomass in the monitoring program. And presently in the monitoring program, only phytoplankton biomass is used because the phytoplankton biomass is normally increasing with increasing nutrient availability. In the present project, we asked the question if we can use taxonomic groups as an index also, and if DNA metabarcoding can be used as a complement to microscopy. So the present project organization is, we have 4 different work packages. Work package 1 is to develop different methods. Work package 2 is to optimize metabarcoding procedures. Work package 3 is to harmonize reference data. Work package 4 is actually to compare metabarcoding and microscopy. We have an advisory committee consisting of international experts. We will contribute to reference databases. We will contribute to Swedish Oceanographic Data Center, and we have outreach activity. So I will first talk a little bit about the work package 1, which is about methods. So during the past years that has gone, we have collected 340 or 350 samples in Swedish marine waters. You can see the sampling points there in the map. We have tested, or we are testing different storage temperature when we do the filtration in the sea, from the seawater. We have figured out what DNA extraction method that we should use. And we have also used spike-in DNA or synthetic DNAs to normalize. We do that to every sample to normalize the method and to get some sort of semi-quantitative analysis of the DNA. We also take samples to compare the method variability between microscopy and DNA, and we test filtration volume. So presently we are doing sequencing at the NGI, short read metabarcoding sequencing using Illumina. And we are also planning and got into the work with long read metabarcoding using PacBio. Yes, and with this, I would like to hand over the word to Meike.
Hey.
Yes.
Welcome, Meike.
Thank you.
So.
Excellent. So I'm Meike Latz. I'm working as a postdoc on this project, and I'm working towards optimizing metabarcoding for monitoring of eukaryotic plankton with a focus on primary selection, mostly so far. For phytoplankton, the most commonly targeted gene for metabarcoding is the 18S rRNA gene, because it contains both conserved sites, which are shared across species, and are therefore very good targets for primers, but also variable regions, which are the regions that we are targeting to get taxonomic information. And in this project, we selected 10 primer pairs that we want to test for short read metabarcoding using the classic Illumina MiSeq approach. And 2 of them are primer pairs that we newly designed in this project, but we're also performing long read metabarcoding using PacBio sequencing that's targeting almost the whole rRNA operon. And there we have designed also new primer pairs on the 28SG. Just to give a quick overview of how we design new primer pairs. So we use sequences from a database. For example, for 18S, we use all the sequences from the PR2 database. Then we cluster the sequences to get rid of overrepresented sequences and use the centroid sequences of the clusters and align them. And based on this alignment, we can use a program called dgPrime to design degenerate primers with a degeneracy of 12, what we used in our project, on every position of the alignment. And then this program also outputs the number of sequences that are matched by the primer in each of the positions. And thereby we can select primers that seem very suitable because they match a lot of sequences. Then to first evaluate the primer pairs that we selected, 10 primer pairs again, and 2 of them are new. The other ones are published. They're here named by the first author name in the publication. And when we look at which primer is most suitable for our purpose, we first look at the amplicon length distribution. And this is very important because we are limited by Illumina MiSeq sequencing to around 500 base pairs, and therefore we need the forward and reverse reads to overlap. So primers that exceed that length are not suitable, but also we want to have a highest as possible length because longer amplicons provide more taxonomic information. As we can see in the taxonomic resolution plot, the V9 amplicons that are only about 180 base pairs, they have much less taxonomic resolution than the V4 amplicons. Then it is obviously our very important point is the coverage on different taxonomic groups. And in the gray bars, that's the number of sequences from the database that are matched by the primer. And it also gives an idea of which taxonomic groups here on phylum level are matched. And for example, here we can see that the V4 primers, they're not very good in matching the excavate groups. And that's a disadvantage, but we need to make a compromise. And our compromise was to select the Balzano primers because they performed quite well in silico and also in the in vitro result that I'm presenting next. So to evaluate these 10 primer pairs further, we prepared libraries and sequenced those libraries on samples from 3 different locations, one in the North Sea that was kindly provided by Uwe John, and one from the Kattegat and one from the Bothnian Bay. And we have 2 replicates for each of those locations. And when we look at the NMDS plot, where we plot all the samples with all primer pairs, we can see there's a strong clustering based on location, so that the bias introduced by the primers is much smaller than the effect of location. And when we separate these plots, the NMDS plots based on location, there we can see a clustering of the samples based on the region targeted. We see a separation of the V9 and V4 primers. The Shannon index below in the box plots provides an idea of the in-sample diversity. And here again, the primary most important for the diversity is still the sample origin, but also the primers will introduce some bias there. and also the community in the bar plots below, we get a different impression of the community composition depending on which primers to use. So we selected the Balzano primers because they have a good performance both in silico and in vitro, and they're also used by many researchers, so their data is comparable, which is very valuable. And the new primers that we designed for the 18S gene are not performing better, so it makes more sense to use a published primer pair. The next steps will be to compare these results with long-read sequencing results, where we will hopefully get much more taxonomic information and higher taxonomic resolution, but also with shotgun metagenomic sequencing, where we get like an unbiased view on the community composition because we're using unbiased primers. Thank you. Next will Bengt Karlsson present some reference data information.
Okay.
Yes.
Please, Bengt.
Okay, so I'm Bengt Karlsson, working at SMHI in Gothenburg, Oceanographic Research. So one thing we want to do with all the data we gather here is actually to use it in a sensible way. So to do that, we need to harmonize reference databases and data storage. And we should keep in mind that the reference databases are actually updated continuously. There's new information coming into the reference databases. And of course, the data should be open access. And we also think that the data should be available at the national data host for marine biology and oceanography. The SWAM and the Swedish EPA, they designate these national data hosts for monitoring data in Sweden. And of course, we keep in mind that the data is part of an international effort. Now, let's see. So, what are we doing now? First, we are proposing to SWAM, the Swedish Agency for Marine and Water Management, a new investigation type, a ny undersökningstyp for plankton barcoding. And this is based on this sequencing of 18S and 16S. And from the samples from we're collecting now from this year, a full year of sampling. And the idea to have it in this database is, of course, that then we can use it together with the older data and with all the environmental parameters, such as salinity, temperature, nutrients, and so on. And the reference databases we are using is PR2, the Protist reference database. That's for the unicellular eukaryotes. In fact, the phytoplankton and the microzooplankton in the sea. And for cyanobacteria, we use the 16S gene in the SILVA database. But to be able to use this together with the traditional data from microscopy, there is one list of organisms or phytoplankton for the Baltic Sea, the HELCOM Phytoplankton Expert Group list. And then there's another one for the Kattegat, Skagerrak, and the Norwegian Sea, which is called the NOMP list. And then we have these global phytoplankton or algae lists, the Algae Base and the World Register of Marine Species. So just to give one example, there are of course many diatoms in the sea, and we only work with the planktonic ones. And there's one genus called Zudunichia. There are roughly 50 species described of Zudunichia. And in the light microscope, they are a bit similar. They look like these canoes that you see in the picture to the left. But looking down the transmission electron microscope or the scanning electron microscope, you see these morphological details that discriminate the species. But in the Proteus reference database 2, we found 10 submissions of this species, Zudonychia delicatissima. 5 of these were unique. So comparing the microscopy data with the molecular data is something we will be working with next year. So to summarize, the next steps include analyzing the results from Illumina and PacBio sequencing and compare the metabarcoding results with microscopy, and also to analyze the results together with the environmental parameters. We have a special focus on the harmful algae. And then, of course, we really try to establish metabarcoding as a new tool for phytoplankton monitoring. And we think it will complement microscopy-based methods. And it is cost and labor efficient, especially if you handle very many samples. Then the metabarcoding method is quite efficient. And the results include information on biodiversity, non-indigenous species, harmful algae, and so on. And we can expect changes in biodiversity due to climate change, of course. Okay, thank you for your interest.
Thank you, Bengt, and thank you also to Maike and to Agneta. We will now continue with the questions that you have asked in Mentimeter. So I will ask Torbjörn Ekrem, Christer Erseus, Maria Karlert, Agneta Andersson, Maike Latz och Bengt Karlsson to please put on your microphone and your video so that we can ask you questions. Or maybe you can stay unmuted but have your microphone ready and put on your video so that we can see you. And we also have now Gunilla Eidung with us. Welcome, Gunilla. And you will ask the questions to the speakers that we have gotten.
Yes, I will. Hello, everyone. I have no sound, do I?
Now there's the sound.
Okay.
Yeah.
Hello, everyone. And thanks for very interesting presentations. The first question goes to Torbjörn. Can information available in barcode libraries be used to reliably assess intraspecific genetic variation?
Excuse me, it's maybe only me who is hearing you very poorly.
Yeah, I'm also hearing very poorly. The sound is really, really hard, bad.
So the sound for Gunilla here is not perfect. I will repeat the question. It's for Torbjörn Ekrem. Can information available in barcode libraries be used to reliably assess intraspecific genetic variation? There's a question from Robert.
Yes, I guess it's a very good question, and I think the answer depends on a lot of conditions. For instance, if you trust the identifications that have been made, and if you have enough sequences of that particular species in the reference library. But let's say that you have a nice representation of different populations of that particular species. And maybe you're a taxonomic specialist of the group, and you could reliably approve of the identification that's in there. Then barcode information gives you a lot of extra data on intraspecific genetic variation throughout the whole distribution range, if that is sampled. And we've done that kind of studies with chironomids a couple of times. For instance, in a transatlantic fashion, looking at the genetic distances in populations distributed of the same species distributed in North America, Greenland, and Northern Norway.
Thank you. Let's try again, Gunilla. What question would you like to ask?
Okay, there's another question for Torbjörn. Can you hear me properly?
It's still a bit rusty, but
it's pretty poor.
Okay.
I think it's the mic at your.
So maybe you can hold it to your mouth.
Like that? Will that be better?
No. So yeah, it's perfect.
Okay. So, Torbjörn, are there museums that store DNA extracts as vouchers? Would be nice for future studies, but research takes space and energy. And this one is from Morten.
Okay, so I will.
I'll just quickly repeat that question because I think it was
Yeah,
not so easy to hear.
I will repeat the question.
How to deposit and store DNA extracts. And I think we're looking at two different things here. One is to store the DNA extract from the voucher that was used to generate the reference barcode. And that should definitely be stored, but at the same time, it's a limited resource, right? So if you use that same extract for sequencing different markers and doing, let's say, phylogenetic studies and things like that, it will run out eventually. So then there will not be any DNA voucher left. And then the second question, or the second aspect of this could be, what about keeping a DNA voucher of the bulk sample or the eDNA sample that has been collected in a monitoring program, for instance? And I think that also is a very important aspect of it. We should definitely keep DNA extracts of bulk samples and eDNA samples that are being made and keep them, store them in natural history collections. There are natural history collections that do this today, but there are also research institutes that keep those samples safe in -80 freezers, for instance. And the reason for keeping them will, of course, be that as technology develops and more both molecular tools and bioinformatic tools become available, the same samples can be analyzed over again, let's say in 20 years' time, and with a comparison of contemporary samples that are taken from the same localities.
Thank you. While our technicians are working on the sound, I would like to ask Christer and Maria, shouldn't we do full mitochondrial sequences instead of choosing barcode markers? Maybe Christer can start.
Shouldn't we do?
Shouldn't we do full mitochondrial sequences instead of choosing barcode markers?
Well, it depends on what. If you want to use them at barcodes, I think that you can still use barcode markers as long as that is cheaper than doing the whole mitochondrial genome. But the mitochondrial genome may give you other information that can be useful. The problem with species delimitation is not that to have more of the mitochondrial genome, it's to have some independent information from the nuclear genome, so that you can tell something about the coalescence of the different genes. That will give you more information about the species. But for the identification aspect, once you know the species, I think that the resolution of a single marker might be as good as a complete mitochondrial genome.
Maria?
Yeah, because I have an additional aspect regarding diatoms. At least from my experience, the better species separation, or at least the one I've worked with, maybe I should say, but at least the better species delimitation is coming from the chloroplast. And you could, of course, do the whole chloroplast then instead of the whole mitochondrium. But also here, as long as it is working, I think the barcode is okay. But I have asked once if we can't use the whole genome, and then people just laughed at me because it will. Because then you have all the genetic information, but it needs so much bioinformatics and data storage, and a barcode is such a small ID. It's much more convenient to use that one, and as soon so for the diatoms it wouldn't help with the mitochondrion. We need the chloroplast, but yes, in the case of the diatoms, I would like to have a bit longer than with the possible to analyze with the MiSeq. But if the PacBio method works, we get a part of the barcode that is sufficient to separate species. The bigger problem is for the diatoms that we need to have the morphological analysis in place as well, because the taxonomy is still unsolved for many species.
Okay, thank you. We have a question to Agneta. Are there any downsides to using the PacBio long read sequencing over Illumina costs, for example?
Maybe Meike can answer.
Yes.
Given in aspect
Meike?
of this.
Yeah.
It's certainly more expensive, and maybe very rare species within the sample will not be detected. That depends on how deep we sequence, but with Illumina MiSeq, it's very cheap and to sequence that deeply. But also the analysis of the data. There's no established pipeline for analyzing the long read data so far. And it's very handy with the short read data. We have some pipelines and we can analyze the data within, I don't know, half a day. So I'm looking forward to play with that.
Yeah. Thank
maybe
you.
I could add, just add here that there are reference data for the short read sequences available now. They are not yet available for the long read sequences, but that's a matter of time.
Okay, thank you. And then we have a question to all speakers. What would it cost to cover all needed Swedish species for meaningful monitoring using eDNA? That's a big question. Anyone who would like to try to answer that?
Are they talking about all species, or can we talk about the tax at a time, for example, fish species, or do they mean that we need to get the reference for all the species?
Please do. Michaela.
Yeah, because I think, for example, if you go for the fish species and we need a good reference database, is that what they're asking for, references for all of them? Yeah. I actually think that you could, to have a really proper one with all the fishes, I think that you could get away with not even £1 million. But then the other ones are like so many different taxon groups because you don't have that many fish species. And I even think you can go lower than that. And especially if the different even groups in this consortium can collaborate, I actually think that we could, you know, get the more common ones and also get all the fishes really properly, because there are collections we could work together and do these mitochondrial genomes that we then can use as references for the markers we have.
Thank you. Anyone else who would like to try to answer that question?
Maybe I can give a perspective of what we've done in Norway. The first 5 years of funding from NORBOL resulted in about a reference library of 21,500 species and cost about 100 million Norwegian crowns, roughly. So, but that's including all the in-kind funding and all the external funding, everyone's work through the budget. So it's a yes, you can divide it by the number of species. So there, but it's, you know, it's not for free. But I think what we have benefited extremely from is that people are so interested and so enthusiastic and think this is such a nice idea. And they get extra data on their favorite specimens that they are very happy to contribute.
Yes.
Even what we call you amateurs, which are, you know, really high-end specialists in their groups, are happy to contribute.
But Torbjörn, you are not including the microbiota, are you?
No, we have only included multicellular specimens so far. We had a small test run on diatoms that was successful. I think a fairly high success rate running through our pipeline. but we will try to include that in Bioscan Norway, the next stage, if we get that funded.
Okay, Maria, do you have any comments?
Yeah, I have a comment. We're trying because we don't think that we can complete the diagrams of all Sweden without a lot of effort, and so European experts have decided to work together on a diatom barcode reference database and try to put everything that we get together. Yes, you won't get the regional interspecies differences, or maybe not all of them, but our aim is to get at least one sequence for each species. And the nice thing is with this collaboration, which is this diet barcode database, is that we get both the morphological expertise and the molecular expertise into one working group. So we hope in that way that we can contribute to complete the databases as far as possible. The only problem is that this is a European collaboration, which is not really funded by Europe, because they say that's a national problem with the reference databases. So, but yeah, we continue.
Mm. Thank you. We will now have to go to lunch. And before that, I thank you, all of you who were presenting here on this morning, and all the questions that you have answered. And we will just check that if you haven't got the link to these discussion groups, you should have gotten a mail yesterday with links and all of the information. In that email, there was also a link to the website for this conference, and there you should have the links. But we have now also put in the YouTube web chat the link to these links. Very meta here. So please check there if you can. And if you don't see it, then contact us. We have 2 people working on this, so we will put up an email during lunch here, and then we can send you all these links.
Sarah, did you put the updated one for the standardization?
We will check that. So, yeah. Thank you. And we will have lunch for 45 minutes, and we will be back here at 1 Swedish time. So 45 minutes, and see you back at 1 o'clock. Welcome back from lunch. I hope you had enjoyed your food and that you have moved about a bit so that you don't sit all day. We are now going to look at or listen to the rest of the research project from the funding. We have 5 more projects, and I just want to remind you of menti.com, that you can go in and ask your questions. After the 5 presentations, we will have a collective question time as before. First up is Björn Lindahl. Björn is professor at the Department of Soil and Environment at the Swedish University of Agricultural Science. Björn will tell us about monitoring of biodiversity in forest soils, sampling and DNA analysis. Welcome, Björn.
Thank you very much. I hope you can see my presentation now.
Yep.
So we are running a project that focuses mainly on methodological development of DNA-based methods to monitor biodiversity in forest soils. We have already got quite far in this area. We have a national monitoring program running for soil fungi, but we now try to tackle some challenges with the methods and the logistics to streamline the methods. This is the team. We have expertise on fungi, prokaryotes, and soil fauna, and we're also working closely connected with the Swedish Forest Soil Inventory, headed by Johan Stendahl. And it's Leticia who has done most of the job. We actually got our sequences a few weeks ago, so I will show you very preliminary data. We have primarily focused on soil fungi so far, and because we are working for long in this area, and we know that many fungi form very large mycelia. Here you see a meter of moss folded away from a stone, and you can see all this is one fungal individual. Here you see a smaller individual, and it's trying to defend itself against the bigger brother here, or bigger antagonist, by producing a wall of toxic material. Probably the big mycelium will quite soon conquer this area. So there's a lot of battle going on here between fungi below ground, and as they try to dominate the soil, and this creates spatial variation and makes it problematic when we want to sample fungal community, because we have to take soil sample with a traditional corer, which is much smaller than the individuals that we try to sample. So if you compare with sampling plankton, as we already heard of, you collect water and you get all the individuals in your sample. But here we actually take samples that are much smaller than the individuals, some of the individuals in the community. This is our graphic hypothesis of how we think it looks like in the soil, even on a quite big scale, like 20 meters. We have areas where single individuals of fungi strongly dominate. Although there is, of course, some overlap. And if we take a sample here, we would be lucky and get something that resembles a community where many different species overlap. But if we sample here, we get almost a monodominance by a single species. So, of course, we then have to take a lot of samples, but this is logistically demanding. And if we're going to use this method in national monitoring of soil biodiversity, we need to develop protocols that are time efficient and cost efficient. So we had 12 test area in this project. We sampled young forests, plantations, and more old forest. We sampled nutrient-rich spruce forest and more poor pine forest to get some contrast in the material. All in all, 12 sites. And at every site, we did a quite complex spatial sampling design with triangles so that we could get pairwise combinations of samples with different distance in between them. So we assessed variation from 1 decimeter up to 20 meters with quite high replication. And at each point here, we sampled 2 soil cores, one set that were pooled directly in the field into one bag to get the composite sample of the whole site, and one set where the cores were kept individual. This is how it could look like, a lot of triangles out in the field. Here is Leticia sampling in a pine forest, quite easy location here, flat and nice. Some of the sites were not so easy to implement our nice spatial design in. Reality is always a bit more complex than your plans, but it went okay in the end. And then we took all the samples, extracted DNA, amplify the ITS2 region, which is becoming a kind of standard barcode for fungi, and we use PacBio sequencing. We focused on the organic topsoil of the core, so we removed the moss layer and the mineral soil. And I will present some data, very preliminary on soil fauna, but then most will be on the fungal communities. We'll look at the spatial variation. We also studied the post-sampling storage, how quickly got the samples into the freezer, because this is a major challenge out in the field. And we also looked into pooling, grinding, homogenization. Starting with the soil fauna, here is we're just testing out different protocols. We tested some different primer combinations. It's the CO1, that's the marker. And we're quite successful in getting a lot of animal sequences. We can identify here. We got the Collembola that we know are very common in forest soils. We get some Malacostraca. I'm not so much into animals, but I guess these are crayfish related. Probably they are like wood lice or something. We didn't get good matches. We got a lot of snails and things. Maybe surprising is that a big part of the community was actually from flying insects. This could be dead animals in the soil. It could be larvae. It could be excrements like eDNA. We don't know. But it turns out that if you want to do an inventory of flying insects, the soil is not a very bad place to take samples. Also doing a multi-variational, what's it called? Multi-dimensional scaling, ordination of the data. We could use the animal data and get a quite good separation into our ecotypes with the old forest separated from the young forest and the pine forest from the spruce forest. So it seems to be a useful way forward. We will work more with it. What about the spatial variation in the fungal communities then? Here we got a very nice data, I have to say. For every increase in spatial scale, we got a significant increase in dissimilarity, the Bray-Curtis index. So dissimilarity of 1 means that we don't have any common species in the communities. And you see here, when we compare the different forest types, old spruce with young pine and so on, we are very dissimilar fungal community. But then for every step we take in our spatial scale here, we get more and more similar communities. Of course, when we sample within the forest type, the community gets more similar. Then the only step where we don't have a significant difference is between the 20 and 10 meter, like here and here. So that's where we have a kind of homogeneous community between the 10 and 20 meter scale. So we have some ideas what explain this different variation on the different spatial scales. We have sampled a lot of other environmental parameters like water content, the topology, mineral content, root density, and also fungal biomass. And most of these parameters seems to vary and be patchy at the decimeter to 1 meter scale, which you can understand if you look at how a typical forest soil looks like with these boulders and depressions. And so here we think, here is most of our environmental variation. And of course, on the higher scale, we have ecological variation, difference in ecosystems, fertility gradients, and so on. But we're really interested in this intermediate scale where we have this very clear structuring of the fungal community at a scale from 1 meter to 10 meter. And here we really think that this is variation created by competitive interaction among individuals in the fungal community. And we will look further into that. I think we will get some ideas when we look into which fungal species that drive this pattern. And then we will also look at the bacterial communities, the soil fauna, and also measure some biogeochemical parameters, enzyme activities, to see how they match onto this spatial variation in the fungal community. And we have this idea that the fungi are really creating spatial variation in the other communities and also in the biogeochemical cycling in these forests. So methodologically, this is challenging because if we look into the current methods for inventorying forest soils, we are working around the meter scale, a typical soil science pit. Whereas if we really want to describe the fungal communities, at least, we need to go up in scale and sample at least on the 10 to 20 meter scale. More in the similar spatial scales as the trees, really. If we go into the storage, not surprising, we took the composites storage, technical duplicates, and either froze them directly on the same day as sampling, or we'll let them stand for 5 or 15 days room temperature. And the fungal community changed quite a lot. We have been looking a little bit into the sequences here, and it seems it's mainly 3 different fungal taxa, 3 species with bad matches to any references, so we don't really know what they are. That increases a lot from a few percent of the DNA pool to perhaps 25%. But there are also different other changes in the fungal community. So it seems it is quite critical to get the samples frozen immediately upon sampling. And with the inventory team being out in the field for a very long period, this is challenging. We have to find new practical solutions. Maybe we can get some used freezing car from somewhere. Grinding and homogenization is something we put a lot of effort into, thinking that we sample from a maybe 10 by 10 or 20 by 20 meter forest area into a bag. And then we have to, from this material, select maybe 100 milligram of material from which we extract DNA. And we want the extracted DNA to represent the original forest plot. It's a lot of grinding and homogenization required, but this is very expensive actually and takes a lot of time. It's the most laborful work in the whole process. So if we can streamline this, it would be nice. So here we compared samples where we sampled 50 milliliter, freeze-dried it, and then either took samples directly for DNA extraction, or we did another ball milling step where we grinded the material to fine powder and then sample for DNA extraction. And we took pairs of the samples all the way through, and our expectation was, and this was just to confirm that if we would take samples directly, there would be very much more variation in community composition, and that our homogenization is really necessary to get replicates to be similar. Little bit surprisingly, already the pairs taken from this granular material with chunks of millimeter-sized chunks were quite similar in the technical replicates. Maybe here is a bit dissimilar, but in general they are as similar as the samples that were finely grinded. What is interesting is that there is a systematic shift in community composition due to the grinding, and probably there are some fungal species that have more recalcitrant structures or resting spores that really needs this bead beating to release the DNA. We need to take more samples and check this out before I dare to recommend that we give up the ball milling and go directly for sampling of the original material. finally, look into different strategies of pooling. Here we compared either pooling directly in the field, where all the cores were mixed in a bag, and then we took one composite sample for sequencing. As an alternative, we kept all the cores separate, extracted DNA from all of them, and pooled the material post-sequencing, like bioinformatic pooling. One would think that the above method, where we keep all the samples separate through the sequencing, would keep more of the information in the material. But in fact, we found the opposite. Here you can see ordination of the different samples, different sites in color, and they nicely group according to their ecology, with young spruce, young pine, old pine. But then when we look at our samples here, where the red ones are pooled into a composite samples already in the field, and the green ones are kept separate through the sequencing, we see that we actually lose ecological signal by keeping the samples separate. The composite samples are further out in the ordination and more separated according to their ecology, whereas all the separate samples, which are pooled bioinformatically, move to the middle and become more similar. And thinking a little bit about this, we can come up with a plausible explanation that there are very many generalist fungi that have quite low biomass but occur in high frequency of the samples and also across the different ecological forest types. And then we have other species that are more of a C strategist, highly competitive. They're likely to form very high biomass but very localized. So maybe these species are only present in a few of these cores, but with a very high biomass. And we suspect that these C strategist species that are competitive and locally dominant maybe have tighter ecological niches and are more informative when it comes to the ecosystems. And these hotspot species, if you want, actually, when we keep the sample separate through sequencing and pool relative abundances data-wise, they will be diluted by all the low biomass samples here. Whereas if we sample the soil directly and pool it in the field, these high biomass species will actually have a stronger signal in the composite sample, and their ecological information will be better preserved. So
I
think we're not only
break in here and say that it's time to sum up.
Last slide. So here we're saving a lot of work, and we're also preserving the ecological information better by pooling the soil directly. I want to flag a recent paper by Carles Castaño. There has been some discussion comparing different sequencing technique. This is a paper comparing PacBio sequencing and Illumina sequencing for ITS markers, but it's applicable to any markers that have some size variation. We really find that Illumina does not work here, and PacBio does a much better job. So check this up if you're interested. So we are getting closer and closer to our dream machine, where we actually can nicely get out fungal communities and hopefully communities of other organisms just by putting soil samples through the machinery. Thank you much for your attention.
Thank you, Björn. We will go to the next second speaker. We will sum up with questions after these speakers, as I said. And our next speaker is Francisco Nascimento. He's an associate professor at the Department of Ecology, Environment and Plant Sciences at the Stockholm University, and he is at the Baltic Sea Center. Francisco will tell us about nematode barcoding, advancing monitoring of Baltic benthic ecosystems. Welcome, Francisco. Let's see if we have Francisco with us. We see your presentation. Do we have sound?
So,
So,
can you hear me now?
Yeah, we hear you. Perfect.
Yeah, okay, thank you, Sarah, for the introduction. So, as Sarah mentioned, I'm an associate professor at the Department of Ecology, Environment and Plant Sciences at Stockholm University. And I'll be talking to you today about nematode barcoding, our project that tries to advance monitoring of Baltic-Benthic ecosystems. So we're working a lot with the Baltic, and as many of you know, it's a heavily affected system by anthropogenic pressures, both by old pressures like eutrophication and oxygen deficiency, but are also expected to interact with climate change and new challenges. So we need to monitor to detect and predict the effects of these pressures and manage the risk of these pressures in marine ecosystems, and particularly in the Baltic. And this is a big part, an important part of HAF's mission. Benthic ecosystems, the seafloor, are severely affected by all these pressures I just mentioned, and they have been monitored traditionally by focusing on macrofauna. Macrofauna are invertebrates that are larger than 1 millimeter, and they are monitored by HAV once a year in a large spatial scaling effort that tries to detect changes in this community. This data is then used to predict and assess habitat quality, benthic habitat quality, and uses tools that have been developed for fully marine systems. However, as you know, the Baltic Sea is a brackish system, and these tools do not fit completely to what the Baltic's brackish systems need. So here in this graph, you see the BQI, which is an index that classifies habitat status. And you see that here is the salinity on the x-axis, and it correlates greatly with salinity and species richness. So, so this means that for areas of the Baltic Sea proper around, for example, the Stockholm archipelago area, there are very few stations that can be classified over moderate, and this has management implications. This has mostly to do with the naturally low diversity of macro-benthic species in the Baltic. But Baltic-benthic ecosystems are not species poor in terms of animals, not if we consider also the smaller invertebrates that we call meiofauna. So there are invertebrates between 40 and 1,000 microns, and they are heavily dominated by nematodes. So nematodes are the rival insects in terms of the most diverse animal group in our planet. And so we think, at least in the Baltic Sea, we have around 200 species of this, of nematodes. We actually did a first large-scale meta-barcoding survey of nematode diversity, and we see that perhaps this 200 species is a conservative estimate. So here on the S-axis, you see our sampling effort going from northern Baltic proper in the Stockholm area down to the Arkona base. And there are two things that it's good to highlight here. So you see a lot of colors. So we have identified a lot, 61 nematode genera. Another thing that I would like to highlight here is the black colors of these bars here that you see. These correspond to unclassified nematodes. And this indicates that we don't have good enough reference databases. And we have heard this morning a lot about the importance of these reference databases. So nematodes can be a powerful bioindicator of environmental change, so it would be a good candidate to monitor if it was not so cumbersome to sample. I mean, they have low mobility or sessile, so they have a practical time frame of response to disturbance, and they have a highly diverse community, which includes species with different sensitivities to anthropogenic pressure. So our central aim of this project is then to try and use this information to improve benthic monitoring in the Baltic Sea and the tools that are used today to classify habitat status. And to achieve this, we have two central aims. So one of them is to improve the reference databases for Baltic nematodes, to decrease those black bars of nematodes we cannot classify, and then to contribute to improve monitoring tools applying DNA metabarcoding. So I can give just a short update or short status of the work we've done so far. So we have sampled 90 stations in 2019 and 2020 that cover the area between the Botnian Bay and the Southern Baltic Sea proper. From these, we have isolated nematodes from the sediments, not working with eDNA, but with community DNA in this case. So we have isolated these animals from 116 samples and tested optimization for DNA extraction for protocols for nematodes. So regarding the first central aim of this project, to improve the reference databases for Baltic nematodes. So far we have advanced quite a bit in the last year. So we have identified 115 nematode species from the Baltic Sea, covering all this span of both latitude and salinity. And these have been amplified for the whole 18S marker, plus the D2, D3 region of the 20S marker that has been also suggested to be a good marker for species delimitation in nematodes. So all this, we have now DNA extracted and sent for sequencing from these 115 nematodes, and we should be able to upload this data to reference databases beginning of next year. We have also done quite a bit of work to try and identify different taxa that can be used as bioindicators for the particular pressures that affect the Baltic Sea. So one of them is oxygen deficiency. And here I would like to talk to you about a study where we have sampled sediments along an oxygen deficiency gradient. So from fully oxic that we see on station A to hypoxic and oxic, differing in quantities of hydrogen sulfide, which you know are very toxic to these animals.
I'll break in here and say that you have a couple of minutes left.
Okay. Then here we see that we find quite a bit of animals living in anoxic sediments, in hypoxic sediment also that can be used as bioindicators for oxygen deficiency. We did the same for organic matter enrichment, another proxy for eutrophication. Here we see that communities with low organic enrichment separate quite nicely from high organic enrichment due to the physical and biochemical characteristics of the sediments. And here we can identify here also the community composition and try to see which taxa are important to differentiate these the responses to this. So the next step will be, it's already ongoing work, is to try and couple sequencing data to the biomass and abundance of key nematode taxa that we have determined that have different sensitivities to these pressures we have identified. And then we can try to relate these differences in biomass in nematode species with different sensitivities, how they vary spatially from the Botnian Bay to the Southern Baltic, and temporally. We have now a 4 to 5 year database where we can test these questions also in a temporal scale. This is just initial data from shotgun sequencing that we see that spatial difference seems to be a lot more important than temporal difference. And then, of course, the last step is to try and incorporate this. If we have biomass in abundance of these species, we can then start incorporating this information on biotic indexes that can help us to classify habitat status. And perhaps in the future, I think one of the advantages of working with eDNA is that you can monitor multiple communities simultaneously. So why not expanding this approach to include bacteria and archaea and their potential to perform ecosystem processes that are vital for the functioning of benthic ecosystems. So this is something we're very interested about, looking at the potential of bacteria to perform vital processes in benthic ecosystems. And with this, I would like to thank my co-applicants and collaborators that were involved in this project, and of course, Naturvårdsverket and HaVs for their funding. Thank you very much.
Thank you, Francisco. Very interesting. And we'll go to our third speaker. We have Per Sundberg. He's professor at the Department of Marine Sciences at the Gothenburg University. And Francisco has stopped sharing, and Per will tell us about eDNA in environmental monitoring and biodiversity assessments, remaining issues. Please welcome Per Sundberg.
Oh, thank you very much. Yes, this project is about remaining issues and the use of eDNA in monitoring and biodiversity assessment. It's besides myself, it's Alice Axberg, Thomas Dahlgren, Mattias Obst, and Marina Panova, and we are all in the Department of Marine Sciences at University of Gothenburg. This is one part of the 3-year project funded by the EPA, and it started in 2019. There are plenty of really success stories about when it comes to eDNA and environmental monitoring, especially when it comes to fish. But we reckon in the group that there are many more remaining questions before we know how and where we can actually use eDNA in environmental monitoring. And this project is not taxon orientated or methodologically orientated. It's more into questions about this. So what some issues that we have recognized are, for example, do all phyla release eDNA at a detectable level? And how does it compare to traditional biodiversity inventories? And how long does DNA stay in the environment? And how far away from the source can we detect DNA? And often people ask, well, is it possible to estimate the population size from eDNA data? And of course, one of the final aims of this project is to develop protocols and recommendations to stakeholders. I should also mention that we have collaboration with Aarhus University, Department of Biosciences, and GEANS, which is a EU North Sea Interreg project, which actually works with building a reference database for marine invertebrates. We also get data from ARMS Embryon, which is about reef monitoring structures in international projects. So, for example, what about all animals? Do they actually release eDNA at a detectable level? If you want to, I mean, we talked about a lot of these studies have been on fish. Fish swims around and probably leaves a lot of traces of DNA in the water. And I must say, we are. Sorry, I should have mentioned that we work on animals, and we are all zoologists, and we also are in the aquatic environment. But when it comes to marine environment, there are 30 phyla there. Of course, these animals, they are different sizes, they have a different ecology. So what about polychaetes, bryozoans, riverworms? Do they actually leave traces that we can see in the water? So what we've done in this experiment is that we have taken samples from public aquaria where we kind of know the species composition. In this case, we're doing eDNA metabarcoding using 18S, CO1, and also 12S as a fish marker. I should mention straight away that we have big problems with getting, due to COVID-19, a lot of these experiments or analyses has to stop. We have to wait for especially extraction kits, and we hopefully to do this now. They promise now they can deliver the extraction kits now in December. But the samples are taken, and we took one sample, or not one, but several samples in one aquarium up in Channon Marine Laboratory, which is an open system. Of course, we also take the samples from the incoming water, and we also have taken samples from the Universium in Gothenburg, which is a closed system, where we also have known the historical what has been there in that system. So this is now under analysis. We're using here a meta-barcoding approach to this particular question. Connected to this, we have also how does eDNA compare to traditional biodiversity inventories? And we have two studies, ongoing studies in the marine environment and focusing on benthos. And we have done parallel sampling for eDNA water samples and also traditional techniques. Once again, we're using here the same markers, 18S, CO1, and 12S. The first one is about hard bottom reefs around coast of Denmark, and this is a joint project with Aarhus University Department of Biosciences. They have taken around 10 sites along the coast of Denmark, the east and western coast, taking samples there, and at the same time we have taken eDNA samples, water samples. We also done two inventories in two sheltered bays in Sweden on the Swedish west coast, and there we used we will compare our results with inventories done by two very experienced consultancy companies doing this kind of inventories. Is Marine Monitoring and Medins here in Gothenburg. All the species lists here are now finished and ready, but we, as I said before, we're waiting for the extractions kits to do the comparison. This is going on. So this will tell us if we will have the same result as those there. Will we have more species? And I should, referring to what Ekrem told this morning, that we must be aware of that when it comes to the marine environment, there is really for the marine invertebrates, there isn't really a database, a reference database. I've seen estimates of maybe 20–30% of marine invertebrates are actually sequenced. So we will never get a full comparison, a list of species. What we can see is, which Torbjörn was talking about before, we may see diversity, but we will not be able to say that this particular species or this sequence is this particular species. Another question which we found very important is how long does eDNA stays in the environment after the source has disappeared? And this is also connected to the risk of false positives, which is we consider a general question. For example, if you take a sample in a lake where there has been a bird dropping, it's been feeding another lake, will we actually pick up that particular species in that lake, or even though it's not there? We also in our group we've been engaged in port surveys and ballast water. And for example, if there is a ship there emptying its ballast water tanks in the port with DNA from another port, but still there, and we take a sample at the same time, we will find this invasive species. We will get a false positive in that. So this is an important question: how long will it actually stay there after the source has disappeared? Two laboratory experiments there. One is done by Thomas Dahlgren in our group. It was on the cold water coral. They kept the animals in the tank for 24 hours, and then they were removed. And then DNA was measured from filtered water samples and analyzed using droplet PCR. And what you can see here is that quite quickly, after a few days, it goes down quite radically, although there is still remains a long time ago. But this is quite important. I think Björn mentioned this as well, the importance of actually deal with the sample straight away. You cannot wait for a few days because a lot of the DNA is actually not detectable at that stage. We're also doing a similar experiment right now with round goby, which is an invasive species. We have to keep them in tank for 24 hours. We measure the DNA from filtered water samples, We have various replicates here and numbers of fish. And then we have, by predefined water time intervals, we actually measure the DNA. This is ongoing. We were halfway through this when we couldn't get more extraction kits. And once again, we are using droplet PCR to do the actual management here. Or sorry, the measurements. I should also mention this is a collaboration with people in the biology department in Gothenburg, which have the required permits to work with a vertebrate species like this. We also, in this experiment, we've also combined this with we want to see, we'll weigh the fish and see if there is a correlation relationship between the number of DNA reads and the weight or the biomass of the fish. And this down here, you can see what this droplet PCR looks like. These are positive droplets here, indicating that there are this particular species in the water, but also it can be quantified. The number of DNA reads can be quantified. So we want now to correlate that to biomass and the number. Was this yes, as you should mention, what we're also using the round gobe as we look, because this is an invasive species. So we've done some sampling around the port of Gothenburg to see if we can develop a monitoring program for this invasive species. And in this connection, I would like to mention a challenge here is if we have a very sensitive technique picking up DNA in the water, the question remains, what do we actually sample? What area are we sampling? If it can traverse a long way, if you go to a bay, see if there's a fish there, the species is there, and we can have a positive response, but where does it stay there, or could it be that it comes from another area? So this is something to consider when you interpret the results. Another question which is I often get is how can you is it possible to estimate the population size from DNA reads? And we have one we're using freshwater pearl mussel as a model for this because we've been working on that before. Right now we have oh sorry I'm going too fast here. How far away from the source can we detect DNA? This the population size come next. So we're using freshwater pearl mussel, and we have various small rivers around Gothenburg area where we measure. We know that there have been inventories about this mussel, and we take samples downstreams from these in these rivers. Once again, we use here target species analysis using a droplet PCR. Also in this hard bottom reef study I mentioned before, we had taken samples at the reef, upstream and downstream, so we can get an indication of the differences between upstreams and downstreams and at the reefs. Sorry. Yeah, sorry, this is the population estimate. So once again, freshwater pearl mussel. We now, it has a collaboration with the Skellefteå Kommun where they're going to do in the spring here. They're going to do diving inventories, and we also will arrange with simultaneous sampling of DNA so you can see the correlation between the number of what they can count and the number of DNA reads. We also have one site where we have gone back to see what happens with abiotic variables, how it actually influences the DNA reads. And for example, you can see here this this structure here, and this is from a small river close to Gothenburg. This is you can see how much lower the water is, and of course this is a place where we know there are mussels here, and the number of reads is a huge difference. Of course, there's a lot of water here. You see this structure here. This is the same place. And this is low water, so this is something you have to consider when you're going to do this correlation. What actually are the abiotic factors here? I think that's quite an important issue to think about.
Okay, I'm going to break in here. You have a couple of minutes more.
Yes, great, fine. So upcoming sub-projects, a model we want to look into more of the probability of finding rare species, minimizing the risk of false negatives. How to do that? We had a statistician in the group from the beginning, but unfortunately had to leave, and we're now advertising for a replacement. But this, I think, this is an important question: how to look into sampling strategies, how to actually give some kind of confidence in your results. We will also continue the experiments to estimate how far from the source we can detect DNA by using cage experiments. And we will also do further empirical tests on population size estimates. And finally, what I would like to say that our deliverables here are, of course, there are papers published in peer-reviewed journals, in popular science magazines and reports, but also workshops and protocols, recommendations for eDNA multispecies and target species analysis in environmental monitoring, and also to be able to give guidelines for the best practice for for data management and publications and surveys. And I would say we can follow the project here on our website. It's on the department's website. So that was it.
Thank you. Thank you, Per. Thank you very much. Our next speaker, we have the fourth speaker in the row. We will welcome Antti Vasamägi. Antti is professor of fish biology, and we find him at the Department of Aquatic Resources at the Swedish University of Agricultural Science. Antti will tell us about the applicability of eDNA for monitoring pike abundance in Sweden. Welcome, Antti.
Thank you, and thank you for introduction, and good afternoon. Per Sundberg already gave a very nice introduction to the potential applicability of environmental DNA and a little bit wider perspective in terms of what the eDNA can be used for. And it's not only the interspecific diversity and taxonomy that we are kind of asking here, but there are also other questions important, and one of it is that can we use the environmental DNA to actually estimate species abundance, and this project actually focuses but only on a single species, so it differs from other projects funded by this really great initiative. And so why do we want to identify pike abundance using environmental DNA? And so there are several reasons for that. One is that pike is ecologically and economically important species, but at the same time, it's very hard to monitor pike stocks or abundance. So it would be really nice if we can do it using the eDNA. And with this project, we really try to go into the details and to how can we do it, what are the difficulties, and how well the eDNA abundance actually reflects the pike abundance. As a startup, so now I'm going through some of the ongoing works and then describe a little bit about the future work as well. So as a startup, we wanted to test that how well actually our molecular methods kind of result on biomass indicators using a very simplified situation. So basically we sampled the juvenile pikes and kept them in aquariums at different densities, and then evaluated eDNA abundance prior to this kind of introduction, during the introduction, and after removal of the pike as well. So similar that Per was discussing. And so we used half a liter of water and a very common extraction kit for environmental DNA. And as a first results, so what we used is a quantitative PCR and a Tachman assay. So it's a kind of mitochondrial DNA fragment of 94 base pairs. And this has been used earlier in environmental eDNA work in pike. And here you can see some of the preliminary results on pike biomass in grams versus the eDNA concentrations in nanograms per microliter during 22, 46, and 70 hours after introduction of pike into the aquarium. And you could see that and in the lower graph, what you can see is the same graph, but basically a log scale of the pike biomass, and on the y-axis there is a CQ value, which represents the qPCR kind of output, which is like on what cycle number the signal was kind of detected. So it means that the smaller the value, the more target DNA you have in a sample. So what we see is that actually the first results were quite promising. So we have a reasonably good relationship between pike biomass and eDNA. So we were quite happy about that. And so the next question, what we were then going to is that, does this reasonably nice relationship hold if we go to the larger scale? So we did this experiment this spring using adult pikes of varying size, less than kilo up to 8 kilo pikes, and then in a kind of open air tanks with a diameter of 3 meters, the tanks consisted of 10 cubic meters of water. We used double filter combinations instead of single filters in order to potentially reduce the clogging issues. So what were the results? Before I go actually to the results, we were also interested on technical details, such as which DNA extraction method actually works the best in our case. And we tested this in 2 different pike densities, high density and low density, and tested 3 different methods: very common blood and tissue kit and power water, and then the kind of very simple method called Cellex 100 resin. And on the y-axis, again, there are CQ values. And what was, to our perhaps surprise, the most simple and cheap method, which is Cellex, worked the best in terms of it gave the highest concentration of pike DNA from the filters and also produced the highest yield altogether. So based on these results, we opted that, okay, we go with the Cellex for the next steps. And here are the very fresh results of the eDNA biomass relationships. On the top graph, you could see the pike biomass on x-axis at 3 different time points, 48, 120, and 168 hours. And on the y-axis, you can see the CQ value. And initially, we were quite disappointed about the results of the undiluted DNA because we got a lot of high technical variation during the qPCR, which indicates that there is some inhibition problems in our samples. And but after dilution it 8 times, actually we were able to get very or quite nice biomass eDNA relationship, as you can see here, at all 3 different time points. And you could also see in the graph, the dotted line is actually the expected relationship between the linear relationship between the biomass and eDNA. So at the end of this experiment, we were pretty happy about the results, and we could conclude that actually our approach seems to be working pretty nicely in this kind of big mesocosm scale. But there is obviously more details to look at. And for example, when we looked, where does the variation comes from? It's obvious from the upper graph that when we look, the filter replicates showed very low variation among them, which means that basically every filter gave us the same answer. And similarly, on the lower graph, you can see the PCR replicates, which also showed low technical variation. So basically what we can kind of take-home message from here is that there seems to be this technical variation related to this relationship seems to be quite small, and most variation what we see in these graphs are probably kind of biological origin. And then jumping quickly, what is still ongoing work and which have been done, which we started this spring, is that we carried out quite extensive field sampling during the spring when the pike was spawning. We sampled 22 locations, shallow bays along the Swedish east coast, and 16 bays were sampled twice. And all this effort was synchronized by another project, REFISC, conducted by Länsstyrelsen in Stockholm, and the reasoning for this is that it really gives us the unique opportunity to compare the results from hook and line sampling and estimation of pike abundance versus the eDNA. And all these filters have been now frozen and stored and will be analyzed in 2021. To think about a little bit further about the details, how actually what is the best way to sample eDNA for these kind of for both biomass estimation or also for the diversity. So we really adopted a very, very thorough approach using the transects. So each bay was sampled in 4 transects, 3 along the coastlines and 1 into this kind of a deep transect you see in the middle of the bay. And each dot here is basically 1 bucket of water that we took 1 liter, which we pooled together and then used it as a pooled sample. So we hope we have a very good kind of general samples available for biomass estimations and for next year. And here you can see some of the pictures and the participants of this, seeing how this pooling of water on the left side looks like. And then in addition, we really try to adopt very simple approaches to sample eDNA, because if this is to be used in the monitoring, the methods has to be simple. So we didn't use any fancy pumps, but really try to kind of use the most cheapest, but at the same as simplest and robust approaches, and including these kind of cartridges that contain already filters inside. So what about future work? We would really like to work more on evaluation of the effect of inhibition, work more on limits of detection, limits of quantification. We would like to do more work on different assays using genomic DNA versus mitochondrial DNA to be able to perhaps identify the signal of spawning and kind of released milt in the water. We would like to compare our qPCR results with the ddPCR, as Per Sundberg was explaining in previous presentation, and also with the metaPAR coding. So we could really see how different approaches perform and how much do they kind of agree on. And finally, which is for the next year, we are going to do temporal monitoring, or eDNA, by comparing the eDNA signal with the fish counter signal in the spawning grounds. And so all this work has been done as a really team effort. So basically we have an ePike team that contains the most important person is Eric Carson, our PhD student that you can see with this kind of a eDNA filtering gun in his hand. And then we have a supervisor committee with Martin, Josephine, and Göran. And we also have Patrick and Ophir as a master student. So it's really a group effort, and we are really looking forward for the next season and really getting the kind of most exciting results of this project. So with this, I will finish. Thank you.
Thank you, Antti. Thank you. Interesting and a nice shotgun there. We will continue to the fifth project. This will be the last report of these 8 projects that got funding. We have Mikaela Hellström. She's a managing director at Mix Research. And she will tell us a bit about Life DNA Aquatic, DNA Aquatic. So, okay. Mikaela, please. Let's see if we have sound.
Can you hear me now?
Yeah, perfect.
Excellent. Okay, my name is Mikaela Hellström, and just like Antti, I'm actually just the messenger here because I'm actually going to present some results. We have got in this project called Life DNA Aquatic that is actually administered through Akvabiota. And we are 7 different research institutions or companies that are working together. And we are 11 members in this team. And actually everybody has their own very specific knowledge, whether it comes from GIS or modeling to these nitty-gritties of the molecular biology. So it's actually a very huge team effort that we have been able to do this. So here are the different work packages we have. I put them in a bit of a different ordering because it's just in the order that we have done our research. So we have actually worked both on through the entire pipeline when it comes to eDNA, that comes with planning before you go out to the field to what you're going to do with the data. And we have done some specific reports that actually are based on the first different points here. So the first report is already handed in to the Swedish EPA. And this is guidelines for field collections, filter types, and eDNA preservatives. And it's both based on our own experiences and also on a big project that now is like ongoing within the DNA Aquanet, where everybody's sharing information. And then the second one is DNA extraction that we also are handing in now. And then we have then been looking at preservatives and what kind of preservatives should we use and how do they behave over time. So within the team, we have actually worked a lot with different preservatives. Here in the shadows, some of us are actually working on a whole range. We actually selected first 3 and then went down to 2 in this study. And then we looked what happens if we extract them after 2 weeks, after 3 months, and after 6 months. And we looked then at 2 marine sites, 2 river sites, 2 wetland sites, and 2 lake sites. And here we also have our, in the left down corner, you can actually see our follower, Henrik Appelgren, who actually has been checking how we are doing and even joined us in the field. And what we could see is that on a large scale, there was hardly no difference. Here we also have 2 different markers, so that's why I have the red boxes. But what we could see is that if we go into a smaller scale and we have like more rare taxa, we could actually see that ethanol was performing better than Longmire. And that's especially true when you come to marine areas and when you have like these really salty environments. So after that, we actually decided in this project to continue with the preservatives we thought were the best. And then we also have like done a little bit of spin-outs because Akvabiota did the work for Nyköping-Åarnas Vattenvårdsförbund 2 years ago. And there we actually collected 18 times 2 samples, and these replicates were put in ethanol. And replicate A was extracted within 2 weeks. So what we did now is that within this project, we extracted the other ones after 470 days. They had been stored in the fridge, and the results were actually identical when we looked at the metabarcoding results. Then we also had another project that actually came in because of an accident, because we were supposed to send some DNA samples that were stored in TE to our team members in Belgium. But as you know, there are a lot of problems with the mail nowadays. So these samples got really delayed. So they were more than 2 weeks in the mail. And now we had a golden opportunity to compare the integrity of the delayed samples with backup DNA samples we have. So the measurements are ongoing. And then here is also a bit of a spin-off. I will just move this one because it's actually the County Administrative Board of Blekinge was working together with Aquabiota to compare test fishing with nets, marine nets, and eDNA. So at each of the 16 stations, there are 3 fishing nets and there's 1 eDNA sample. And just before the nets were put down, we took a sample. And and some of these samples were also like overlapping with the other research that we did within the Life Then Aquatic project. But here we could see that we are definitely catching more species. And then we now have a pretty big project, and now when we know more what we are doing, we also know more what kind of methods we want to use. We have actually been looking at the spatial collection strategies, because there have been a lot of discussions. For example, if you have a big river with high flow, that it actually should be enough to just take water from the river mouth when it meets the sea. And we wanted to check that. So now I need to... So we actually had a few hypotheses we wanted to answer. First of all is that the eDNA signal diminishes when it reaches a new lake or river. Then we wanted to know how far does the signal reach when a species is not present, and also how many technical replicate samples are needed to cover species presence at each locality. And then we also thought of this, that we actually took 66 samples over 121 kilometers. So we thought that if we actually think of a species distribution model, and if we take biological, physical, and chemical factors into consideration, can we then sensibly reduce the number of sampling points? So we actually did, so here are the sites, and here we are in the field. So we actually did first a connectivity chart of the study area. So here are still no species here. So the big circles are actually big lakes, so you can see the different sides of the lakes. The black bars are barriers. And then we had our a priori model, where we looked at the highest elevation, we look at barriers, outlet to the sea, what is above the highest coastline, and then flow speed of the running water and nutrients. And based on that, we collected 6 sites. And this was only to find the full species diversity. Then if we want to get the nitty-gritty information, we might need to look at more lakes. So we actually collected all these red arrows, but these 6 ones are our a priori. Here are the results, which are quite wild, I must say. So this is like a bubble plot. So each column is actually a site, and each row is a species. So this is all of our fish data that we got. And then we get like an overview, so we can see, for example, the yellow round ones are Perca fluviatilis. We can see that they are at many, many, many sites. We can see that some species disappear at other sites, so we get a very quick overview. And then we compare the data that we have collected from sources that included interviews, sport fishing data, official fishing data, net traps, and rod fishing over the last 30 years. And also this river Moelven is very well studied because it has also been used for PhD and master studies. So it's like we thought here we had the most background data we can get to compare. And we actually found 26 species out of the 26 existing species in the areas. And most common ones were perch, pike, roach, bleak, burbot, and dace. And this also fits with historical data. We also found out that if we take 1 technical replicate at each site, we get 83 to 85% of species diversity at a given location. With 2, we get 97%. And this is just like work in progress. So we are working on a very, very big paper here. So this actually has to do with like, here we have like what the data looks like when we put the fishes out as the family, and we actually we are going to tease this out for like distribution data or different groups of fish. So for example, we are going to do the cyprinids separately, the salmonids separately, and so on. And then we come here with the a posteriori with our theory here, and we actually got 26 out of 26 species in the sites that we had selected based on the data of the environment, like heights, lake sizes, river flow, and so on. And now we have the ongoing analysis there. And then we also wanted to know that how far does the signal go? So these yellow arrows are actually so-called signal arrows, where we actually realized, oh, the signal diminishes really quickly. So the one here to the furthest left corner, where it says brown trout 300 meter, there's a lot of brown trout in one of the tributaries. There are no barriers, but they don't go out to the lake next door. And it has to do with that the lake next door is actually full of pike that would eat them if they go there. And then I was also wondering, so I was asking our fish guru in the team, that why does not the pike go and catch the brown trout? But it's because it's like really high speed running water, so the pike is actually happier to be in the lake. But by knowing like the ecology of these fishes, that we could trust our data, we could actually look at the signal distance and realize the signal disappears really quickly if a species is not present. And then we compared this to official data. So here is like electrofishing data from one of the sites. We have done this for 7 sites. I'm only showing one because we don't have so much time. So here is first electrofishing data that's pulled from 2019 and 2020. That is at the same site where we have collected eDNA. So this is what it looks for 2 years electrofishing. This is what it looks for 7 years electrofishing. And then to really look at the electrofishing data, I actually dug out all the electrofishing data in the databases that exist and was looking at what species have they got which year. So here you can see for each year, there are actually 2 electrofishing sites that are 100 meters apart from each other, that like pulled together here for the electrofishing data. And as you can see, we get much more with the eDNA sampling. And that has to do with that electrofishing is more focused on brown trout, salmon, sorry, help, how do I do this? Salmon and European bullhead, right? And also that when we do electrofishing, we go for shallow hard bottom habitats in running water. And species that swim down to the bottom and stay there are the ones that we catch, the other ones we don't. But if we want to look at biodiversity and how many species we have, we should go for eDNA. And the mussel data is still ongoing and in progress. And then the conclusions here I have already talked about. So first of all, we know that if we have different methods from field test to bioinformatics, we get comparable results as long as users are trained and the labs are adapted to eDNA. And then with the sample storage, if we want to like go very long out in the field and we need to we can't access freezers and so on, you can actually, for example, use ethanol. Even if it takes 2 or 3 weeks to get them to the lab and you then put them in the fridge, they're fine for at least more than a year. And then when you're planning the data, and this also comes in our report number 1, it's like when you're planning how to go out and do this work, you really need to know your environment, because it's like I said to a colleague, it's like the same thing when we do any kind of inventories, you need to have like background data and you also need to know how to deal with the data. So the eDNA does not analyze or choose the sites by itself. And so now we are going to continue, and also that we know that the signal of eDNA disappears very quickly if a species is not present, which is actually very valid when it comes to bigger inventories. And then we have the next work package 5. We look at seasonal variation in one marine, one river, one wetland, and one lake site. And we take the samples once a month, and then we want to see that for these different habitats, which season is the best to collect, or what kind of differences do we see over the year. And then we are doing a cost-benefit analysis, where we are looking at the money, time, and the information gained at what is worth traditional methods or eDNA.
I'm breaking in here. We have you have a couple of minutes. Oh, Okay, sorry.
Thank you.
Yeah.
Good
Right on time. Yes.
Yeah.
Thank you, Michaela. I think
Thanks.
we are going to take a 5 minutes break now. We have heard 5 presentations, and after the break, we will get back to you with questions. So after the break, we will have some questions, and you can ask your questions via Mentimeter, menti.com, and see you back in 5 minutes.
Okay.
Okay, now we're back online. We had a couple of minutes of break and we are going to go to the questions. So I call up Björn Lindahl, Francisco Nascimento, Per Sundberg, Antti Vasimägi, and Michaela Hellström. And you can turn on your camera and wait a bit with the sound. And all of you who are not these people, you can disconnect. So we have some questions in the Mentimeter, but the Mentimeter has disappeared. So we need to check and find. There you go. We have again Gunilla with us. Gunilla, we will try you again with the sound and see. We hope this works better this time.
Yes, we do. Can you hear me now?
Yeah.
Okay, great. So the first question is to Björn. Since DNA is relatively stable, why samples are affected so much by frozen? What is the best way to handle the samples?
The reason why they are affected is probably more because some fungi grow if you store the sample wet and outcompete other fungi. So I think it's more a biological process than a matter of DNA preservation. So we haven't really tested different methods to kind of chemically stabilize the samples, but maybe someone mentioned just soaking the soil samples in ethanol would do the trick.
Okay. Actually, I have a question to you too, and that has to do with freezing. You said you had to find out new practical solutions that you can use in the field. What are you doing? Have you contacted anyone or are you trying to figure out something yourself?
We need to look more into this. It could be that there are just a few very specific fungal species or bacteria when we look into that grow after sampling and that we can remove the data bioinformatically. And after that, the kind of resident community will still be there, quite preserved. That would be the most simple solution. Maybe it can also be treated statistically just by including the number of days between sampling and freezing as a statistical covariate. Otherwise, I think we need to think about easy ways for preservation. But when we're talking about the Swedish soil inventory, the inventory teams are out in the field in quite remote locations. So, I mean, it's not such an easy problem to solve.
Okay, thank you. Francisco, how do you plan to estimate biomass from eDNA data?
Yes, so that's a good question. I went a bit too quickly over that. So we're creating mock communities from this bioindicator species. We know react and have different sensitivity to these pressures and oxygen deficiency and eutrophication. So we will create these mock communities with known biomass and known abundance, and we will try to relate our sequencing data both from metabarcoding and shotgun sequencing to these data and see how they correlate. So we'll be following up on some of the work already done for insects and macroinvertebrates that have fairly good relationships between sequencing data and biomass. More biomass than abundance.
Okay, thank you. Per, why use droplet PCR in the experiments to check for DNA stability? Would it not be more relevant to use the same methodology as for the standard eDNA monitoring?
Could you please repeat the last part?
Would it not be more relevant to use the same methodology as for the standard eDNA monitoring?
Sorry, what methodology?
Well, the same methodology as for the standard eDNA monitoring.
Oh, okay. I see what you mean. Okay, I think there are two different questions you have to ask in monitoring programs. One is, do I want to have a full, complete species survey, and then you use metabarcoding, or you can ask, I'm looking for a particular species using qPCR or droplet PCR. And of course, so this was you could have done it with, but I mean, we start with, in those experiments, we start with clean water. So to do a metabarcoding study, using I'm not sure, it's much easier to use a droplet or qPCR to actually quantify the number of reads. So basically, I would say, when you plan for a monitoring study, you have to ask a question: are you looking for a particular species? Or are you looking for a species list of species?
Okay, thank you. Antti, does the source of the DNA matter, as from same individual fecal DNA with inhibitors, mucus or other sources? Will that affect the time the DNA lasts in the water or the breakdown speed?
I'm not sure if I got the question exactly. So can you repeat the question?
Yes.
Well.
Do you have problem hearing me?
Yes.
So you hear
Okay.
Sarah better.
Yeah,
Maybe
so
she should
I will
read it.
repeat the questions. Does the source of the DNA matter, as from the same individual fecal DNA with inhibitors, mucus, or the sources? Will that affect the time the DNA lasts in the water or breakdown speed?
Yes, it's a great question. So the question, is it is the DNA comes from the feces or from the mucus, so the expectation is that if it comes from the mucus, it's kind of the DNA, it's less degraded. So that means that it may stay longer in a system, but it really depends on many things, so biotic factors, temperature, water movement. So so I think that at this point we really don't know what is the relative contribution of feces versus mucus for pike, for example. We actually were planning to do some experiments on well-fed pike versus not fed pike, but we ran into some small difficulties that the pikes didn't want to eat anything, so we don't have the data on that.
Okay, thank you. Michaela, do you put the filters in ethanol? Would you recommend that for phytoplankton on filters too?
I actually think that you have to do a test with that as well, because now we have been working with like amphibians, mussels and fish. I actually didn't have time to even present the amphibian data, but I think when it comes to phytoplankton, that it actually needs to be tested.
Okay.
But I mean, it could definitely work, but I don't dare to say anything before that one is tested. Mm-hmm. But it could be quite quick to do a test where you actually and if anybody wants to test that one out, I'm more than happy also to share the way that we set up the experiment for different preservatives. So it can be done very quickly.
Mm-hmm. I also have a question to you. You said that you can get comparable results as long as users are trained in labs that are adapted to DNA. But that is maybe not always the case. Do you have any ideas of how this could be a reality in order also to increase the quality of monitoring?
Yes, what I actually think is also that a very important thing is to train people how to collect the samples. Because that is actually like the most important, to collect and actually fixate the sample as quickly as possible. And then when it comes to the labs, I think that we will have to have some kind of ring tests and actually like help each other out to like live up to the conditions for the labs that are actually, so you don't get contamination, which is very, very easy to get. But I would say that the main thing would be that, for example, let's say when we take water samples for water chemistry, so there are a lot of people are trained to collect the water samples, but then they send them to different labs. So I think it's like the most important thing is to learn how to collect them, and then also to like have these ring tests and also these like certification on labs so that they can actually fulfill these conditions that are needed.
Mm-hmm.
Does that sound feasible?
Yes.
And maybe like have training workshops and so on.
Yeah, but based on what you say, I would say then citizen science wouldn't be possible to use here.
It is actually, it's a bit hard to, yeah, the thing is that it depends on your requirements, because I would say that I would put the requirements in like 3 different points. That one is that if you're using the data, for example, for hydropower, for like really big restoration decisions and so on, then you need to have like trained people who are collecting it. Then we have like scientific research, which is important, but it doesn't financially and practically maybe affect so many people. And then when you come to citizen science, at least you can get like the number of species you are getting and maybe taking some negative controls, but you just need to think of what you're going to use them for, because I believe that citizen science could be really important, and it could be very nice, for example, to involve schools and so on. And then people need to be a bit trained, and then you just need to put the limits on what you can use the data for. But I think you can definitely get some kind of overall picture of that. So it is just on different levels. But yes, I would very much like to be involved in a citizen science project.
Mm-hmm.
Mm. Okay. We have a raised hand in the room as well. Antti, you would like to say something?
Yeah, I would just wanted to kind of add to what Mikaela was saying, that it really depends on the question. I mean, that's what, you know, how useful is the data sampled by, let's say, citizen, I mean, whatever, students or pupils or, yeah, anybody. So it really depends. And it's kind of a, one cannot say that how useful or useless it is. It really, you have to look at a specific question, I think. And species presence absence is probably, in most cases, the easiest answer, but at least in shallow, small water bodies.
Mm-hmm. Thank you.
And I could, can I just add also quickly to that one is that that it's the same thing when we do any other kind of work. For example, when we have student projects and the students are the first year at university or the gymnasiums, they can practice and they can do things and get results, but we maybe don't trust those results for decision making. So I think this is like the same level.
Okay.
Okay.
Thank you very much. We will continue in our program. Thank you all to all you speakers, and thank you for answering our questions. We have gotten a lot of questions, so we couldn't take all of them. But we will now continue, as I said, and we will give you now a bit information about these breakout groups that you are going to be discussing in. And we hope that most of you have know where you're going, what you made your application to when you signed up for this conference. And there is a link again in the chat of the YouTube channel, so you can check it out there if you don't have the link already. You should have gotten a mail yesterday with a personal invitation to the room you have chosen for the conference. No, that's not true. I'm sorry, but in the email you should have gotten links to the different rooms anyway. The first room is standardization. The second room is data handling and open data. The third room is communication, sharing knowledge from researchers to management and between the Nordic countries. And the fourth is the Nagoya Protocol. When you get into the room, there will be moderators meeting you there. So we would like to please check into the room immediately after we have let you go from this session, so that the moderator can introduce and say hello, and then you can choose on how you want to proceed. You will be in some groups, in some rooms, you will be divided into subgroups because there are so many of you. And so make this clear before, and then you will be able to go to the bathroom or to on a break or something that you need. So it would be really good if you connect directly to this link that you have. We will wait for a couple of minutes because we know it's hard to find all of these emails and links, but we hope that you will find the room that you were looking for. If you don't know where your room is, or what you chose, please choose any kind of room and you will be sorted in there. We also have rooms for mingling. If you haven't chosen a group where you want to attend and discuss, you can go to a mingle room, and this room will be open for you to discuss or to take a coffee or just have someone to chat with. So there we will not have any hosts for the rooms. And we will meet back here at 4:15. So you have, I look at the watch now, it's a bit after 2:30. You have 1 hour and 45 minutes about. So you will have time to discuss, and information and instructions will come from the moderators in the group rooms. And with that, I will let you go out into the discussion groups. And as I said, if you don't find your room, pick one from any of the rooms in the list. See you 4:15. Welcome back. We are now back in sending out from this conference room, and we hope that as many as possible from you in the discussion groups have come back, but we will wait for a couple of minutes to see that everyone is rejoining here in the YouTube channel. We hope that you had good discussions. We have seen that a lot of you are still discussing out there, so we are hoping that we will collect you all and gather you back to this room again. And we want to make a reminder for Menti, menti.com. Go in on Menti and ask your questions, because we are going to have a last presentation. And we will also, I'm now looking at our technical people here, that we are now, we are also going to have on Menti a last question when we stop the conference, that so you can still join after the conference is ending to see what people are saying about the conference. So go in on Menti now, because now we are going to hear our last presenters for the day. We are really happy that so many of you are still with us. We will listen to Maria Jansson from Swedish Agency for Marine and Water Management. All of you who have been wondering what people have been talking in these abbreviations, SWAM, that's the Swedish Agency for Marine and Water Management. And then we have Kristin Lövenborg from Swedish EPA, or SIPA as it says here on the presentation. They will tell us about monitoring intraspecific genetic diversity, pilot work at SWAM and SIPA. And we will start with Kristin. Welcome, Kristin.
Thank you so much for the introduction. Yes, I hope that even though people might be a bit tired this late in the afternoon, that you will last a bit longer for our talk today. We will talk about the work that is going on at our agencies related to development of monitoring of intraspecific genetic variation. Because our agencies have different responsibilities, different areas of responsibilities, we have been working with this a bit separately, but we still have a very close communication about common areas and where we can benefit from cooperation. So we will just start by a brief introduction to why we need monitoring of genetic variation, and from the perspective of the agencies, and what kind of information we want to get from monitoring. And then we will go on talking a bit about the ongoing work at each agency, and finally some words about our work together between the agencies. What is monitored within the different monitoring programs is determined by, of course, many things. For example, demands from international directives can have very strong impact on what is monitored. Also, need for data within management to support and to improve management is very important. And we also have politically decided national environmental objectives. And in Sweden, we have 16 such objectives. And in 7 of them, those that are listed here, it is stated that we are to ensure that species and populations have sufficient genetic variation for long-term survival. And also within this framework, we have a milestone target that is called knowledge about genetic diversity. And it states that we are to map and monitor genetic diversity in Sweden. Environmental monitoring, it's supposed to follow the state of the environment over a long period of time. And this then allows us to detect changes in the environment and take appropriate action in time. And data from the monitoring is very important to support management and political decisions. And we can also use monitoring to follow up on different management and political actions. In the end, then, what we want to use monitoring for is to be able to detect negative changes in the environment so that we can take appropriate action and improve overall state of the environment when it's necessary.
Yes, and so what we need here is we need to know the actual state of the environment when it comes to genetic variation, both within species and within populations. And to have use of that, we need it to be general so that we can compare it over time and over space. So we can compare one side of the country to another, and preferably also one country to another. And we need to have reliable data in all aspects when it comes to standards and quality systems and methods that we can trust. So therefore, it has to be stable and over time. And in the end, we want to have a broad picture of the genetic status, and to get that, we have limited resources from the authorities to monitor the environment. So we have to use the resources wisely and efficiently, and to have the right plan to do that. To explain that, I will draw some parallels with medical healthcare. And that when we come to research in both the field of medicine and in genetics, we've really come far, as you know. What we have done, what is done in previous years, was not possible to imagine before. And although the science field is still developing, the diagnostics, treatments, and knowledge about the human body, along with genetic methods, has really come far, as you know. And as authorities, we don't necessarily need the most complicated front-of-the-line techniques, as I explained in the last slide. But we need something to know the general condition of the environment. And therefore, we need a system to monitor overall health or the overall state of the environment. And since this is a Nordic conference, I tried to make a translation of clinics so everybody understand, but I understood now that it should have needed more languages. But that is vårdcentraler in Swedish, hälsocentra, en sundhetscentra, en terveyskeskuset, en hälsokeskuset. Yeah, sorry for my pronunciation. And in the end, we need to distinguish between who is healthy and who is not healthy, or what is a good state in the environment and what is not a good state. And to draw parallels again with the medical care, we need to know what to look for. And if it would be like at a general practitioner, that could be fever, high blood pressure, or a high or low pulse, for example. and that is our indicators. And then we need equipments to measure that. And if it would be at the GP, that could be a thermometer, a blood pressure monitor, or a watch, for example. And that is our genetic methods. And then we also need to know what is a good value of a healthy patient and what is not. And for us, that is our threshold values. And when we combine all these results from all the patients, we get the overall state of the country. So in 2017 to 2019, a pilot was performed by the researchers Linda Laikre and Kerstin Johannesson and their colleagues. And they were using brown trout and bladderwrack as model organisms. And in this pilot, the methods, indicators, and threshold values was evolved and also chosen from research. And in this process, it was a dialogue between SWAM and the researchers to choose important species for management. And in the end, this was evaluated through SWAM's reference group for marine and freshwater monitoring. And this ended up in a program that was developed together for SWAM and researchers. And for now, SWAM has actually started some pilot monitoring this year and for next year, also conducted by Linda Laikre and Kerstin Johannesson. And now we also have Stefan Palm from Swedish University of Agricultural Science on board. And we do this to test and to evaluate and develop this program from 2019. And 4 species were chosen that were by special interest for several fields in the management. But they are also used, those species are also used as a platform to test the indicators and other components of the program. And the 4 species is herring, and that is what the Swedes eat pickled at every single festive occasion, as a lot of you know. But they are also by concern because of the changed fishing pattern, and that combined with the lack of sufficient knowledge of populations made this especially interested. And then we have cod, and that is the cod is not in a good state in the Baltic and in the Swedish west coast to the Atlantic. Do I need to say more? I think every of you is aware of that. And salmon is by special interest for freshwater management, and due to the altering of hydromorphic conditions in the rivers, or simply said, that could say that not being able to migrate due to hydropower dams that stops them from migrating in the way they should. But it's also an important and popular species for sport fishing, but they are also in a bad condition. And eelgrass has been on decline for long, but is also a very important habitat, especially for juveniles of many species. And I will present you to the indicators in the program. And those are built on scientific guidelines in conservation genetic research, and it has to be seen as a starting point. And in case it's needed, they can be recalculated if we use other indicators. And the indicators used is delta H, and this is a simplification, I should say. And that is change of genetic variation within time, within a population, over time, I mean. And then we have NE, which is effective population size. And we have delta FST, which is change of genetic variation over time between the populations. So those will be used as a starting point, but maybe it's a good guess. And for those indicators, we have threshold values. And I just show this to show you that we do have threshold values. And they should also be seen as a starting point. But you can see that we have general values for a good status, a bad status, and for an alarming state as an alert. And the program itself is that was reported in 2019 and now started in 2020. And the thing you should look especially at is 2020 and 2021. And what we do is that we have in a process within SWAM and with the researchers, we have chosen 12 species, and that is a starting point that surely need to be modified along the way, but it's still a very important starting point. And it works out that it starts with sampling, then analysis, and ending up in a report for species that have a very well-known genetic knowledge about those organisms. And those that are not very well known, they need to start out with an inventory phase to know more about the genetics, and then take some planning, and then they can go into the program. And you can see this is run every third year for the species to start with, but that will surely be modified since due to the lifespan, if they have a short or long lifespan in the end. But we start out like this. And now I will hand over to Christine and SEPA's work on their monitoring program.
Thank you, Maria. Yes, a bit about the work that is going on at this Swedish EPA. It is clear that we are to develop monitoring of genetic variation, but of course we cannot monitor all species. So we need to make some sort of prioritization and look at which species that may be most suitable for monitoring. It's perhaps a simple question to ask. It's simple to ask the question, which species should we use in monitoring? But it's quite difficult to answer this question. So one way that we have looked at it is that we have looked at different groups that may be of particular importance to monitor. And for example, these are just examples of some of the groups that we have that we are looking at. So for example, species that are being hunted or where we have fishing to a large extent, that it may affect the genetic structure of the populations. Also, species that are listed in the different directives are an important group to consider because the monitoring of such species, the data from the monitoring could be used for reporting on status and trends. Another important group that we have looked at is species that are at high risk of unwanted gene flow by large-scale introductions of individuals that have a different genetic background compared to the native populations. And finally, pollinating species are, of course, a very important group that is talked about a lot these days, and the Swedish government has also put in extra funding for developing monitoring of this group, not only genetic monitoring, but monitoring overall of pollinators. But what is also very important for us to consider when we look at how we can develop monitoring is to look at what the preconditions for monitoring are. So the best is, of course, that we can start something quite easy and that it can be also cost effective. So, for example, if we have species where there is available tissue collections, that would make it easier to look at trends over time, even early on in monitoring. Or if we have species where monitoring is already ongoing. So, for example, if there is, it doesn't have to be, of course, sampling for DNA, but it could be other kinds of sampling, or where individuals are being handled, so that it could, we could easily add an extra step to the monitoring by sampling DNA. Also very important for us is that the species that we choose already have developed methods and techniques for DNA analysis, so that we don't have to start all from scratch, because we need the monitoring to be cost effective, and we need to see how we can add into what is really already occurring. So to our help to pinpoint some species that could be fit into these requirements, and that are from important groups, we commissioned a report to a group of researchers, and this report was delivered this year, and it contains suggestions on species that could be suitable to use, and genetic methods, sampling sizes also, and costs for monitoring at 3 different ambition levels. And this table shows the species groups that are represented in this report, and it was important for us to have a broad taxonomic coverage, but also, of course, they had to consider what is available in terms of, for example, genetic methods and so on. And right now we have commissioned 3 pilot projects that have started this year, and these projects are a result from the report that we had delivered this year, and they will give us very important data that we can use in our further work on developing monitoring, and also the data can be directly used into management. And also, since quite a few years now, we have within the large carnivore inventories, we have sampling of DNA, and this is to identify individuals, to be able to estimate population sizes, but the samples have also been used to study the genetics of these species. So for brown bear wolverine and wolf, we have quite good knowledge about their genetics in terms of, for example, genetic variation and gene flow between subpopulations. And also for the Arctic fox, we have since 2 years back started to collect samples for DNA analysis. And but even though there are some things going on and has been for a while, we still need more a program that is more aimed at looking specifically at genetic variation, and we still need to determine what species that are suitable to start with, kind of like SWAM has done with the 4 species that they have started a pilot monitoring for. So that is what we're doing right now. We are looking, using this report and looking at which species that we could start up monitoring for. Yes, and yes, so as I said in the beginning, the Swedish EPA and SWAM we have a very close dialogue on things that we have in common within this monitoring, the development of the monitoring, and we have talked a lot about the need for input from both from research but also from authorities. So, for example, we need to have expert input on choice of indicators, which genetic techniques that we can use, and also threshold values, for example. But we also need to get input on what the needs are from management and directives. Yes, and also we will further on in our work, we will have to evaluate everything to ensure that the program is doing what it's supposed to do. Maria, do you have anything you would like to add?
Not really, but in the end, we need to have a way to find out when we need to respond and to take action if the state is not good. So I guess that that was.
Yes. And also we are discussing, this is an example of areas where we see that it is important for us to cooperate and discuss about. So, for example, threshold values, how we store data, and how we ensure that we have a high quality of our monitoring, and also the need for reference groups, both to get input from research, but also from management is very important for us. Yeah, Maria, do you have something to add here?
No, I think that takes it, and also that we feel like that even if we don't have everything perfectly in place, we have to start to do the monitoring so that we can develop the whole system, because we have, as I said, we have it's like we don't have any clinics at the moment to see the overall state.
Yes.
Thank you. Thank you very much. We have some questions who have come in here, and I would like to ask you, Christine, where are the fungi in the report Absolute Monitoring?
Yes, I know that there are groups missing from the report, but they have. This report will also be published soon, I hope, and then it will be available through our website. But they have looked at many different factors when they have made the priorities. So, for example, groups that could be important for monitoring, and it has, and also these preconditions. So, what is available with regards to genetic techniques, for example, to study intraspecific genetic variation.
We have another question. How can we measure genetic variation of endangered small animals, such as small insects, without damaging their populations by sampling?
Yes, it is possible to do non-invasive, non-destructive sampling by, for example, take just a small fragment of a butterfly wing, for example, without killing the animal. That is possible.
Thank you. And we have a question without, I don't know which one of you can answer this, but are you looking for genetic techniques that will work in many species, or are you developing special methods for each species?
I can answer for SWAM. We will, actually, what will be used at the moment, it will be different techniques depending on what is available and what would be most suitable for each species. And I think you were doing the same at the Swedish EPA.
Yes, it depends on the species. Yes, to a large extent. Yeah.
Okay. Okay, so we have a question to you both. Do you monitor those species by single species monitoring approaches, like filtering, participation, extraction, and amplification step, qPCR, ddPCR? And if yes, do you use a multiplex assay? Could be an idea to reduce costs.
Yeah, that was a quite technical question. Yes. I think maybe we need to pass it on, but I know that we will take samples from individuals for SWAM's monitoring, if that is an answer. And we need to have very detailed techniques to get enough markers and enough genes to really see the detail of the populations, like to have to see local adaptation and connectivity. So if the question was if we're going to use something like eDNA, we're not going to use that because we need further detail at the moment for that.
Christine, is there more information available on these pilot studies?
Yes, I think the easiest way, because they are not finished yet with their work, so the easiest way is to contact me and I can tell more about them, I think.
Perfect. And then we have some privileged people in the chat room or in the Zoom room that we are using that can ask their questions directly. Antti, you flagged that you would like to ask a question.
Thank you very much. Very interesting talk. I was just wondering about, it's a huge work to kind of go through the prioritization and standardization and selection of species. So is there a need of more wider consensus and wider research, involving more researchers in a way into this? Because as I see it now, it's kind of it's a start, and few researchers have kind of put the most effort, but as the list of species are growing, I think that it would be important to have it as wide as possible.
Yeah, so I can start on that. Yes, we have been thinking quite truly at that and SWAM, and we definitely need like a reference group to evaluate the monitoring we're starting. But I think we have really thought about it, and we think it's better to have something to evaluate and to criticize rather than like really thinking too long. So we want some like a starting point so that we can have a reference group to evaluate and modify the program.
Yeah.
Was that an answer?
I have another one if I can, but
yeah, sure.
Maybe Christine thought differently.
No, I agree. We need to start somewhere, and then we definitely need to have some reference group or reference groups, depending on how many, for example, species groups, and if we we will need experts in that particular field. So, but right now it's that we need somewhere to start, just to be able to develop it further on.
And maybe this is more like a forward thinking, that what about more wider kind of monitoring or integrated approach, that, for example, we know that diseases and parasites are very important for some species and kind of their survival of populations. So is there some plans to kind of combine, for example, genetic monitoring with some disease or parasite screening? For example, in Atlantic salmon now, there is a big problem in wild Baltic salmon with this acute disease, and I mean it's kind of I would see at least quite a big need for more integrated analysis of different things that threaten the species. One is genetic diversity, but during these sampling efforts, one could actually get very valuable information about their pathogens and parasites, and which actually are threatening these populations so is there any thoughts along these lines, or you have been working mostly on genetics now and genetic monitoring?
I can answer for the salmon. Actually, SWAM have 2 new monitoring programs starting this year, and one is in those genetics, but we also have a starting point of monitoring of fish health and seafood. And so we haven't really integrated those 2 at the moment, but definitely that could be some reason for thought. And we also strive to integrate the monitoring into other monitoring, if you see what I mean. For example, like we are one of the species we choose, we look for like what do you call it? That is also like a health program looking into I forgot what is it? Miljögifter.
Environmental, yeah.
Environmental toxins. We choose like one of the fish species because of that, for example. That since we already have a program.
Christine, would you like to add something in the end now? We are going to wrap this up.
No, we haven't come as far as SWAM with regard to starting up some kind of pilot monitoring, but absolutely we have thought about, for example, for the pilot project on moose that we have, we would like to be compare the statistics of deceased or animals with like the chronic wasting disease, for example, or other kinds of mortality, and compare it between regions and link it to genetic variation. Yeah. So
thank you. Thank you very much for this presentation of your work and what's happening at the different authorities. We have now like a couple of minutes left for this conference, and there are only I'm gonna give the computer back. We have only one last question for you, and we would like to hear from you in what ways you took something with you from these discussion groups. And with that, we will let it scroll, let it flow after the conference has ended, for you all to see what is being said. You can go to menti.com and you can comment there. And with that, we would like to say thank you for today, and thank you all of the presenters and everyone, all of you who have been listening. We have been at a maximum of 185 people listening in. So it's fantastic how you all have been joined up today. Tomorrow we will start at 9 o'clock, and not 10 o'clock Swedish time, but 9 o'clock Swedish time. So thank you for today, and don't forget to comment what did you find the most valuable from the group discussions for today. Tomorrow we go again 9 o'clock. See you then. Bye bye.